our 2020 GT. Excited to be one of today's talk on cell and gene therapy, and we have lined up a very exciting agenda for today and tomorrow. Kicking off today's session with an introduction to quality and regulatory considerations, we have Casey Klein and Michael Brewer. Casey is our Senior Manager for Regulatory Affairs with more than 15 years of experience in global quality and regulatory affairs experience in life sciences, as overseeing regulatory compliance of technologies developed for cell and therapy applications. Michael is our Director and Global Principal Regulatory Consultant from the Bioproduction Division and serves as the regulatory thought leader and expert across all technology applications within the Bioproduction Division. Before I hand over the mic to Casey and Michael, first, this webinar will be done after the live session and accessible through the same Thermo Scientific that you received in your email. Next, if you have a question for our speaker, please send it through the Q&A box near the bottom of the screen. Our panelists are on standby to take these questions, and we will also be addressing them verbally at the end of this. And last, at the end of our second and third presentations today, we will be inviting you to participate in a poll with us, during which you can also share any questions and feedback that you may have for us. Wireless headset, UV sanitizing box. We will have a total of event. Right, I think everybody is going to start. So without any further ado, please welcome our presenters today. Casey, Michael.
Thank you. So today, Mike and I are going to talk about quality and regulatory considerations in cell and gene therapy, raw material selection and product characterization. Raw used in the manufacturing process, but are not intended to remain in the final product formulation. Some common examples of raw materials include cell culture media, cytokines, serum buffers, and enzymes. Raw materials are important because they can impact the safety, purity, and potency of the final. It's important to note that ancillary or raw materials are different to excipients. Excipients are things that are intended to remain in the final product, but it's also good to know that sometimes raw materials can become unintended excipients and still end up in the final product. So here we have the most common raw material guidances for cell and gene therapy across the different. To note that while the guiding are similar across all of the regions, there are some differences. So for example, we know Japan is very particular with the amount of traceability information required for human and animal origin materials. And we also know that in other regions, it's possible to have raw material master files. So for example, in the United States and Canada, you can have raw material master files, which help facilitate review of raw materials by the regulatory agency. But in other regions like Europe, they don't have such a mechanism for raw materials, and you may have to have some kind of customer-facing regulatory support file to support your customers. So now we'll talk a little bit about animal origin definitions. At the moment, there is no regulatory definition to describe the presence or absence of human or animal origin materials in a raw material, but there is a very good recent ISO guidance document, which is ISO 20399. It's titled Ancillary Materials Present During the Production of Cellular Therapeutics. What's good about this is that it gives two levels: the product level, which means there could be animal materials within the product, and the production level, which means there could be animal origin materials used somewhere in the process. So if we compare those definitions to some common raw materials, I will try to classify them according to these definitions. So something like fetal bovine serum is not animal origin free at a product level or a production level, but something like recombinant human insulin can definitely be described as animal origin free at a product level, but maybe not at the production level. So in this example, trypsin was used in the production process for the human insulin, thereby making it not animal origin free at the production level. So we've got a very similar situation here with TGF-beta expressed in CHO cells. Again, that could be described as animal origin free at a product level, but not at a production level. And for chemically defined cell culture media, you can describe this as both animal origin free at a product and production level, particularly if it's made in a facility that claims animal origin freedom. So thinking about those definitions, I've got a case study here called Is It Animal Origin Free? Just to outline some of those definitions in practical terms. So we have a cell culture media that is promoted as animal origin free at a product level. It contains a recombinant human growth factor, and the host cell for that is E. coli, and it's cultured under animal origin free conditions. The growth factor is then purified using a heparin cephalose chromatography column, and the heparin used in that column is derived from pork intestines. So this product is not animal origin free at that production level. Some of the things we need to do, the risk mitigation activities. You need to look at the country of origin of the porcine material. This is important because there are some disease outbreaks of significance. So for example, at the moment there is an outbreak of African swine fever, and it is impacting the import and export of porcine raw materials. You also need to look at the animal health status. Were the materials derived from healthy pigs that had passed both ante-mortem and post-mortem inspection? Has there been any adventitious agent testing performed on those porcine materials, or any kind of inactivation process, for example, heat, treatment, pH, gamma irradiation, or other kinds of inactivation occurrence? And then finally, you need to look at the grade of the material and whether or not master file or some other kind of documentation to help support your risk assessment. So in this particular example, the regulatory authority was satisfied with all of the risk mitigation activities, and the media was able to be successfully used in clinical manufacturing. So now thinking a little more on those sourcing considerations for biological raw materials, we know the aim is to generally avoid animal origin components when possible. But if avoidance is not possible, a risk-based approach for the selection of raw materials is important. So the things to consider include the country of origin. Again, this is important for disease outbreaks, like I mentioned before, but it's also important for BSE, TSE, for ruminant-derived components, but also CJD for human blood-derived products. You want to consider viral inactivation or clearance is possible on your product or process. You'd want to look at the grade of the raw material, whether it's medicinal grade, GMP, USP, or research use. Whereabouts in your process that raw material is used, is it upstream or downstream? You would also want to consider any possible alternatives. For example, could you use a biologically derived protein with a recombinant one? And even with a recombinant one, could you replace an animal cell expression with a microbial expression? And then finally, the traceability documentation available from your supplier to help support your risk assessment. Various supply quality claims that are used to describe product grades or quality levels. So the first one is medicinal or medical device or clinical grade. This means that it is already an approved drug or device, but But it's important to note that you'll be using that product off-label. So it's being used as a raw material. So even though the grade is clinical grade, you still need to qualify that raw material. But generally speaking, these are the most desirable grades, but they're not available for every raw material. Next, we've got pharmacopoeia grade. If a raw material has a monograph, this is a way that a user can use the monograph to show that the material is of sufficient quality for use in drug manufacturing. And a good example would be something like a monograph. Next, we have EDQM, Certificate of Suitability, and this TC, Certificate of Suitability, meaning that if you've got a ruminant-derived component and you've obtained a Certificate of Suitability from EDQM, then that material is of low TSE, BSE risk and is suitable for the use in the manufacturing of medicines. Next, you've got CGMP or GMP grade raw materials. Generally speaking, this does not apply to products themselves. GMP applies to processes. However, it is commonly used for non-regulated products to indicate that the products are made under a robust quality management system. Now, finally, you have research grade, and these are the types of raw materials that are higher risk. They're not intended for further manufacturing, and the suitability of these materials needs to be. So, talking a little more on risk assessment, it is the end user's responsibility to assess the raw materials for suitability in cell and gene therapy manufacturing, and these four main points will help you in the risk assessment. So, first you need to look at source. Like I discussed before, whether it's human, animal, plant, or recombinant. If it is animal, you need to consider whether it was slaughtered or donor animals, and you need to consider the country that it's derived from, particularly around disease freedom. Next, you want to think about manufacturing. Is it a sterile manufacturing process? Is it aseptic, or is it completely non-sterile? Does the facility claim to have dedicated animal origin free facilities, or has it got shared processes and equipment? And does the manufacturer claim to be a GMP facility, and are they self-declaring that GMP, or are they certified to an ISO certificate, or have they got regulatory oversight? So a lot of these types of questions can be answered by auditing. Next, you've got the testing that's associated with your raw material. When you look at your certificate of analysis, is there any virus testing on there? And do you think there should be when compared to the regulation? And if there is any kind of viral risk, is there also any ability to have inactivation or clearance for that raw material? And then finally is traceability. Are you confident that your supplier has the full traceability in their supply chain, and are they able to work with you to get any additional documents or testing results that you may need to help support your risk assessment? My final slide, which is around our cell and gene therapy products, our CTS products. These products are specifically designed for use in cell and gene therapy manufacturing. They're made under GMP, come with extensive testing documentation to help support risk assessments, and we've got evidence of use in over 200 clinical trials now and several commercialized cell and gene therapies. Now I'm going to pass over to Mike, my colleague, and he's going to talk a little more around characterization of cell and gene therapy products. Thank you.
Hello, good day. This is Mike Brewer. I'm the Global Principal Consultant, Director of Regulatory Bioproduction, and I'm happy to be here tonight to talk to you about regulatory perspectives in cell therapy and gene therapy, and specifically some of the applications that Thermo Fisher Scientific can provide you for testing critical quality attributes during the manufacturing process of cell and gene therapy products. A little bit of background, characterization and testing. A variety of tests are required involving characterization, product safety, consistency, manufacturing controls, properties of the treatment. What I'll touch on tonight primarily, we'll talk about adventitious agent testing, residual host cell DNA testing during manufacture of recombinant virus that may be used in either the cell therapy process or as delivery vectors in the gene therapy workflow. And also a couple options we have for potency testing, particularly in the modified CAR T cells. There's regulatory guidance globally for mycoplasma testing. It is a required test at the end of cell expansion in the cell therapy process or at the bioreactor harvest when you're producing recombinant viral vectors, whether those are delivery vectors for a gene therapy treatment itself or a viral vector used for transduction of the T cells to introduce the transgene. So both processes do require mycoplasma testing. Traditionally, mycoplasma test is a 28-day test that's done in cell culture, but due to the limited dating period of many cellular products, it's frequently not feasible for a sponsor to perform the recommended culture-based assay for release testing. In these cases, we recommend the use of polymerase chain reaction or PCR-based mycoplasma assays or another rapid detection assay. So I'll talk about some of that after I finish covering the regulatory requirements. In Asia also, they have similar guidance around mycoplasma testing as a requirement, and the fact that you should use a rapid test should be utilized for cell and gene therapy type products. Some more on the US FDA guidance, and this is quite relevant because it was just recently updated in January of 2020, and many regulatory jurisdictions around the world follow a fair amount or a fairly large amount of the guidance that's published by the US FDA. So regardless of your regulatory jurisdiction, you can anticipate that some of these guidance will be very similar. And primarily here, the recommendation is that product release assays be performed at the manufacturing step necessary where the adventitious agent such as mycoplasma might be, and I'll cover that, I think, in the next slide, after I describe the mycoplasma test that we provide. We are the manufacturers of the MycoSeq mycoplasma detection assay, which is an industry-leading solution for rapid mycoplasma testing based on qPCR. It's designed for lot release testing in a GMP testing environment. It's designed to meet or exceed the guidance that was laid out by the European Pharmacopoeia in 2007 on mycoplasma testing with nucleic acid-based test methods. It's a fully integrated solution that includes sample preparation and protocols for lot release testing application. The instrument comes with an integrated analysis software with features that enable 21 CFR Part 11 compliance or electronic records, and including user access and audit trails and long-term storage of the electronic data. We support our system with instrument IQ and OQ validation services, and we have a global support network of experienced field application scientists that can deliver full workflow training from sample preparation to results interpretation. The assay provides high confidence in results. It's sensitive. It enables validation at levels of 10 genome copies or 10 colony forming units per mil of the test sample. It's specific. It does not detect any non-mycoplasma organisms. The results interpretation are objective and utilize multiple parameters, and again, enabling the highest level of confidence in either a negative test result or a positive test result. And it has full workflow controls as expected for an analytical method that's used in a cGMP environment, including a patented discriminatory positive control molecule, which is unique, and it is a very powerful tool. It eliminates the possibility of a false positive test result if the positive control nucleic acid is accidentally introduced into a test sample. So again, increasing your level of confidence in a test result. Following validation, regulatory filing, and review, our customers received regulatory acceptance to use MicaSeq for lot release testing applications across multiple therapeutic modalities, including cell culture manufacturing, cell therapy, tissue therapy. And this is across multiple regulatory jurisdictions globally, from the US, the FDA, the European Medicines Association in Europe, Japan, the Korea, and we have customers in China and Taiwan and Singapore that are using the assay as well. So here's a diagram that illustrates the typical CAR-T cell therapy manufacturing process, from harvesting of the cells, a selection of the cells, genetic modification of the cells, and at that point the cells are expanded in cell culture, and at the end of that cellular expansion process, typically that is where mycoplasma tests would be performed, and we highlight here in the red, and it's typically performed on the spent media, because that's typically where, if mycoplasma infect the culture and start replicating, they'll primarily be present in the spent media. But we also can enable testing of a small number of the modified T cells in the same test to ensure that if any mycoplasma were associated only with the T cells and not in the media, we would enable detection of them from cell association. Again, it's typically done after the expansion phase at harvest. And for some processes, although it seems to be rare, it may be that you would want to test an additional sample just prior to infusion, and we can provide protocols for that as well. Similarly, this is the recombinant viral vector workflow. And the mycoplasma test in this process is typically done at the bioreactor harvest, or the cell factory harvest used for manufacturing of the recombinant virus. And that's illustrated here. Following that, the cells would be lysed and going through DNA removal or DNA size reduction and the subsequent steps of purification of the recombinant virus. So just illustrating where the mycoplasma test would take place. Now, in residual host cell DNA regulatory guidance, these residual host cell DNA quantities contained in final dosage form must follow the guidance established by local and global regulatory authorities, and these include the FDA, the EMA, the World Health Organization, and local regulatory jurisdictions. It does seem that regulatory jurisdictions globally have arrived and I feel comfortable with the guidance that was published, I think, in 1998 by the World Health Organizations, and that is residual host cell DNA and contained in the final product or in the final dose should be less than 10 nanograms of host cell DNA per therapeutic dose. We have seen some customers that encounter challenges in recombinant AAV manufacturing. And that can be that some of these vectors will package some of the helper plasmid sequences that are used during the transfection process, or some host cell DNA from the mammalian cells that are used to replicate. So our assay can measure the host cell DNA if it's present there, but we can also provide more advanced analytical solutions if you're encountering a challenge with inadvertent package of either vector DNA or the host cell DNA. Some other guidance here, and especially we do want to cover the most relevant and recent guidance. I won't cover the whole slide in detail, but from the US FDA in their guidance that was released in January of this year on human gene therapy investigational new drug applications. They're aligned with the specification limit of host cell DNA of 10 nanograms per dose. And additionally, it's suggested that the DNA size, if present, is below 200 base pairs. And we can provide guidance and advice on analytical methods to determine the size of the host cell DNA that's present in the process. We do recommend that, if you're doing size assessment, that you start doing that relatively early in the process, such as immediately following the DNA size reduction step, which might be a treatment with the enzyme benzonase or DNase. In the adeno-associated virus workflow, again, host cell DNA is assessed during the purification process. And finally, because manufacturing of recombinant AAV is a relatively complex process where either helper virus or multiple plasmids are used to transfect mammalian cells, primarily human embryonic kidney 293, but occasionally HeLa cells. We also can provide support. We have some assays in development for quantitation of some of these plasmids that may be used in the process to ensure that those are cleared during purification. And additionally, we recently released a residual host cell DNA assay that's actually a dual target assay for SF9 production systems that measures both SF9 host cell DNA as well as recombinant baculovirus residual DNA that might be present in the process and to ensure reduction or removal during purification. Now I'm going to move on to some potency testing. Again, regulatory agencies have an guide and expect that a manufacturer of a CAR-T therapy have and implement assays that can assess the potency of the modified CAR-T cells. One of these assays is the Invitrogen PsiQuant LDH cytotoxicity assay. And here, what this assay is actually a co-culture where you introduce the recombinant CAR-T cell into an indicator cell line and incubate those for a period of approximately 16 hours. And you assay these using an LDH assay, which detects cellular damage and cellular destruction induced by your recombinant CAR-T cell. And this is some data here showing the donor cells versus in the percent lysis they can induce compared to an untransduced T cell. The next potency test I'm going to discuss is the Invitrogen cytokine and chemokine 34-plex human procardioplex panel for the Luminex platform. And here again, it's a co-culture of the recombinant T cell with the indicator cell line, CD19 plus K562 cells. After an incubation of 5 hours, the Luminex assay is used to measure specific cytokine expression. And although this slide is very hard to see when you look at it on a computer screen, but you can measure the expression of cytokine levels at low, medium, high, and very high levels for the specific cytokines, 34 in all. to assess the potency of your recombinant CAR T cell. And finally, we'll talk about our overall services that we offer in the bioproduction division at Thermo Fisher Scientific around implementation and regulatory support. We can offer qualification and validation support, training by our field application scientists, but as you implement our analytical tests, we can provide the guidance on validation protocol design, suitable acceptance criteria, systems validation services to make sure your systems are qualified for running our analytical testing methods. On the regulatory support side, we can attend Type C meetings with our customers or other regulatory interactions to discuss the application of the technologies that we can provide our customers. We can respond to regulatory queries during your regulatory reviews for the solutions that you're applying to your manufacturing process that we supply. As Casey mentioned, for jurisdictions that use the drug master file process, we do have these drug master files in place. And then additionally, for the jurisdictions that do not have drug master files, we can provide regulatory support packages to answer specific questions regulators might have as they review your application for approval of your process and product. Additionally, we keep very close tabs on recent new developments in the industry, as well as trying to drive some of the dialogue around acceptance of new technologies, and this could include the MicaSeq test. MicaSeq is the first qPCR-based mycoplasma test that did receive, following validation, received regulatory support or regulatory acceptance for lot release testing, both for recombinant protein manufacturing, monoclonal antibody manufacturing, as well as various cell and tissue therapy processes. And I think there I will wrap up, and I think Casey and I are prepared to answer any questions that you might have.
So I'm ready to take your questions. If you have a question for our speaker, please feel free to send it through the icon. So Casey and Michael, we have two questions here. The first is, can you use qualifications?
Yes, that's a good question. You can use RUO media for cell therapy. Qualifying RUO media that you use. Ideally, try and use medical device or clinical grade raw materials as the least risk. But if you can't source that type of grade, the next best grade is a raw material that's specifically designed for use in cell and gene therapy, like our CTS raw materials. And then the highest risk would be research use.
Thank you, Casey, for the answer. And a second question that has come up, your CTS media GMP?
Yes, they are made according to GMP. So the manufacturing facilities for our media products are registered with the FDA as a medical device manufacturer, and we also have ISO 13485 certification at the site, and we follow all the principles of GMP.
Sorry, I'm not sure if it's just me, but you were fading off. Do you mind repeating the second part of your answer again?
Oh, okay. Sorry about that. Yes, the answer is yes, they are made according to GMP. The manufacturing facilities are registered with the FDA as a medical device manufacturer. Although the products themselves are not devices, we follow the same principles of GMP for the manufacture of those products.
All right, thank you for that. A third question that has come in about and quick and validated. both anaerobic microbes?
Currently, we don't have a solution for testing for specific microorganisms. Across Thermo Fisher, and we do have some qPCR-based tests for specific microorganisms that could be applied to the cellular therapy processes, but typically the regulatory expectation would be a very, very broad-based test for microorganisms. In our R&D side, we are looking at potential applications of next-gen sequencing for rapid, sensitive, and very specific detection and monitoring for growth or replication using next-gen sequencing. But that would be something in the future. It is a good question. We understand the need for rapid sterility or rapid bioburden testing, and we are looking, for potential solutions that could be applied.
Thank you, Michael, for that. I think those are all the questions that we have. I'm just going to, for any other questions that may come in. In the meantime,
I do see a question on the WebEx from Karen Nguyen, and I can answer that question as well. That question is in talking about mycoplasma testing during the cell expansion phase. The question is should it be tested 48 to 72 hours before the harvest? And actually, that's a good question too. And one, you can conduct a mycoplasma test at some point prior to harvest, even as much as 72 hours before. But our recommend for the lot release test would be to test as close to the harvest time as possible. I mean, the test takes about 4 to 5 hours time to result, or test immediately after removal of the cells during the downstream processing following cell expansion. So test immediately after harvest. Given the 5-hour window for obtaining the test result, you'll have a positive, and typically you would have your negative test result before you had formulated the cells into final dosage form.
Thank you, Michael, for that. All right, so there are no other questions that I can see. Is any Yep, okay, there are no other questions that I can see. So thank you again. We will now move on to our second presenter, Dr. Prem Jayaraman. Dr. Jayaraman is our regional field applications scientist for cell and gene therapy. And in this presentation, the seamless transition to commercialization, as well as discuss viral and non-viral delivery approaches used in the genetic of cell and gene therapy applications. Again, for the benefit of those who have just joined us, please feel free to the Q&A box near the bottom of the screen. Now, over to you.
Thanks, Lydia, for the great introduction. Thanks, Casey and Mike, for a great starting session to kick off this Gibco Galaxy Tour webinar. Good morning, everyone, and thanks for joining today's webinar. My name is Prem. I'm the field application scientist for cell and gene therapy in APJ. So I'll be spending the next 40 minutes or so to walk us through the next-gen solutions to accelerate the development of your gene therapies. Okay, so what is gene therapy? To be specific, the gene therapy is the use of genetic modifications by either altering or supplementing a gene that is absent or abnormal in patients, either with the inherited genetic disease and more recently to treat specific cancers and viral infections. So over the last decade, significant advancements have been made in the gene therapy field by delivering the therapeutic gene into the target cells. So gene therapy could be either in vivo or ex vivo based. In vivo based gene therapy, which is depicted on the left side of this picture, basically it refers to modifying any abnormal genes directly in the patient's body. So to do that, we could either use a viral-based vector or non-viral-based vector as a means of delivery. So in the case of viral vectors, safe and modified viruses can carry the gene of interest, such as the AAV, while the non-viral vectors could be either a naked DNA complex and delivered via different carriers, such as the polymers or the peptides shown in pictures. Okay, on the other hand, if you look at the right side of this the patient body, ex vivo based gene therapy, which is the most looked upon right now, which is the gene modified cell therapy, for example, the CAR T therapy, involves isolation of target cells from patients, genetically modified them ex vivo outside the patient body, followed by expansion in the lab prior to reinfusion back into the patient. Okay, let's look into the recent cell and gene therapy global landscape. The pie chart on the left shows the latest distribution of different viral-based vectors used in gene and gene-modified cell therapy clinical trials. So a large chunk of this pie were taken up by the viral vector-based methods. However, non-viral-based methods are also picking up speed in parallel. So on the right, you can see that more than 1,500 phase 1 and 2 clinical trials happening individually and ongoing around the world. So cell therapy, which is inclusive of gene-modified cell therapy, largely outweighs the current gene therapy trials. But with the latest approvals, gene therapy for different diseases are picking up a linear growth phase. So in the recent years, the number of global CAR T clinical trials has dramatically increased. As you can see, a fourfold increase in the last 4 years, from June of 2016 to June of 2020. It's not surprising that almost 59% of the total clinical trials are ongoing in the academic settings, while the rest of about 41% of the clinical trials are taking place in large industrial centers. Okay, in this slide, as shown here is this schematic of a typical gene-modified T-cell therapy workflow. And we have products that support every step of the workflow. But in this talk, I'll be focusing only on the cell engineering delivery systems, such as the viral-based lentivirus and AAV, and non-viral-based electroporation using gene editing tools, such as the TALENs and the CRISPR. Okay, in this section, I'll be talking about our innovative Gibco CTS LVMAX lentivirus production system. So in the case of lentiviral production challenges that manifest themselves in terms of scalable and cost-effective lentiviral production. So on the left, I would like to highlight the current challenges that are associated with adjuvant-based HEC293 cell culture for lentiviral-based production. So the challenges are kind of manual, labor-intensive, and low productivity, and thus it's not a scalable option to larger volumes. And adjuvant-based culture system includes serum-containing media, which poses a significant safety risk and a lot-to-lot variability issues. This would significantly add cost associated with it. So to address these problems of scalability and cost-effective lentiviral production, we have previously developed and launched the CTS LVMAX lentiviral production system. And this is a scalable, high-density suspension system that generates high titers, as you can see, 1 or greater than 1 10 to the 8 titering units per mil as xeno-free components and with all the necessary regulatory support along with that. And the reagents are all manufactured under CGMP guidelines. So if you look into the components of the LVMAX system, what really makes the LVMAX system special is the way that all of these components have been developed to work synergistically with each other, so that to allow for significantly enhanced titers of the lentivirus. So starting with the viral production cells, a high-density suspension adapted cell line that generates high titers of lentivirus. So this cell line is expanded in a chemically defined growth and production medium, and the supplement as well. Along with that, a robust, high-efficiency transfection reagent and with the optimized LV packaging plasmids. And top of this is a novel production enhancer, which enhances the production of lentivirus are added together in the cultures to further drive the lentivirus production. And the result of all of these components working together synergistically is often 10 times or more lentivirus compared to comparable lentiviral production systems in the market. Okay, just to give you an example, one of the key components of the CTS LVMAX system is our viral production cells. And these cells are derived from HEC293 cells. As I mentioned earlier, these cells are suspension adapted in a chemically defined medium and can grow in very high densities, significantly greater than 10 million cells per mil in a standard shape flask or a stir tank bioreactor system. So these cells are free of SV40 large T antigen and are banked under CGMP guidelines. So on the right side of the slide, you can see a number of testing that are carried out for CTS viral production cells versus what is typically recorded for the RUO version, such as assessing only the viability post-thaw of the cells or bacteriostatic or fungistatic activity, as well as mycoplasma testing. On the CTS version, you can see at least 14 categories here that really cover the safety and quality of the cells that are going to be the vital part of your viral production, viral vector production process. Okay, so all of these components that I mentioned are made to work together in a very streamlined production workflow. It's very simple to be performed, and typically for the LVMAX system, cells are prepared 1 day before transfection. So the cells are seeded usually at around 3.5 million cells per mil. And these cells are allowed to grow overnight so that they will overgrow the 4 million cells per mil requirement. And on the day 0, which is on the transfection day, we would take the high density cells, add some fresh media to dilute them back to the required 4 million cells per mil. And on top of that, we would add the 5% LVMAX supplement to the cultures. So the plasmid DNA comprising of your LV transfer plasmid and packaging plasmid mix is diluted in optimum medium. And then this media is made up of, it's a 5% of the transfection volume of the culture. And similarly, the transfection reagent is diluted in the optimum at 5% of the transfection volume of cells to be transfected. So the diluted transfection reagent and the diluted plasmid DNA are added together and complexed for 10 to 30 minutes, at which time they are added to transfect the cells, followed by step 7, which is the addition of the enhancer. And then approximately 48 to 55 hours later, just 2 days later, it is tend to be the optimal time for harvesting the lentivirus from the cell culture supernatant. Okay, so these improved titers of the LVMAX system can be seen when we compare them to traditional methods that utilizes PEI as a transfection reagent. So in comparing to LVMAX suspension system to an adjuvant-based PEI-based process, we can see on a volumetric basis of a 15-fold improvement in lentiviral titers. And compared to your PEI-based suspension process, we would see about a 10-fold improvement in lentiviral titers. So all of these improvements in titers are also translatable to efficiencies in the downstream processing as well, as one would be processing significantly less culture supernatant in order to get the same amount of viral titers. So taking into account the increase of titers as well as other efficiency of LVMAX process, the LVMAX system can save up to at least 50% of the cost compared to your PEI-based lentiviral production methods. And the LVMAX system has also been demonstrated to be very scalable. The innovative LVMAX lentiviral production system that allows you to seamlessly and efficiently scale up your LV vector production with high titers in a xeno-free system. So in a nutshell, you can screen and optimize your lentiviral titers using multi-well plates at the start and efficiently scale up the optimized combination starting from a shake flask to stir tank bioreactors. So as you can see from the graph on the right side, it shows there is no statistical significance between the lentiviral titers using a GFP control plasmid among the 3 different production formats. So the 96-well deep well block plate, which typically would have about 1 mil to a 1.2 mil, to a 125 mil shake flask, which typically has a volumes of 30 to 50 mil, all the way up to a 2-liter stir tank bioreactor, ranging from about half a liter to a 2-liter production volume. Okay, in this slide, I will talk a bit about transferring the shake flask protocols from the LVMAX system into a stir tank bioreactor process. It's a fairly simple process, as you can see. It's similar to the shake flask protocol, where cells and the components are quite robust and easily adaptable into the stir tank bioreactors. To start off with, we recommend to seed the cells at 1 million cells per mil, viral production cells, 3 days before your transfection. But this can be cut down to 2 or even a day by seeding cells at a higher cell density, as shown before. So again, cells in step 2 will be overgrown compared to the 4 million cells per mil target. So at day 0, at the time of transfection, cells would be diluted back to 4 million cells per mil. And just as in the shake flask protocol, the plasmid DNA is diluted in the CDS optimum medium, as well as the transfection reagent. So these are combined together in step 5 and added either by pumping into the reactor directly or via gravity and feed to transfect the cells in step 7. And for simplicity in step 7, what we have done here is like we recommend to add the LVMAX enhancer and the supplement together as a single solution and then pump them directly into the bioreactor, which is approximately 4 hours after the transfection process. So once again, similar to the shake flask protocol, the lentivirus is harvested after 48 to 55 hours of post-transfection. Okay, on the left of the slide, we have shown a readout of some of the key conditions for the stir tank bioreactors that we carried out for the LVMAX, CTS LVMAX system. So here, we had used a 3-liter high-performer glass bioreactor running off a G3 lab controller. Again, the seeding density was 0.5 to 1 million cells per mil. But as I mentioned earlier, it can be higher as 2 to 3 million cells before the day. So the volume of the reactor we tested was about 2 liters. So once all the reagents were added together, and the DO was maintained at a 40% throughout a macro drill hole sparge, and the stir speed was kept at 140 RPM, equating to an impeller speed of about 4.5 watt per meter square. So the headspace air was maintained at 50 mil per minute, and the pH for growth was found to be optimal at 7.25, and this was controlled through pumping in CO2. Whereas the pH for transfection of the cells tend to express and produce the lentivirus better at a lower pH, typically about 6.8. So at the time of diluting the cells down to the starting concentrate, transfection cell density of 4 million cells per mil was around 6 million cells per mil. So the plasmid DNA concentration was used at 2.5 mg per liter to transfect in a 3:2 packaging transfer vector ratio. And the transfection reagent at 6 mils per minute final volume, and the enhancer and the supplement at 4% and 5% final volume, 4 hours post-transfection, with the harvest at 48 to 55 hours, as mentioned earlier. So in the top middle of the slide, you can see the viable cell density as solid lines and viability as dotted lines. The shake flask is shown in light blue line and the 3-liter stir tank bioreactor in the dark blue line. So clearly, it's evident to see the profiles of the growth of the cells, both in the shake flask and the stir tank bioreactors before and post-transfection are quite comparable, showing a similar growth dynamics. At day 0, the cell density was about 6 million cells, where you can see the cutback to 4 million cells per mil by diluting in fresh media. And you can see that the growth profiles takes off post-transfection over the next 48 to 55 hours during the lentiviral production run. On the right-hand side, you can see the comparability of the GFP lentiviral titers as shown in titer units per mil, as determined by HD 1080 infectivity assay, where the 3-liter stir tank is clearly equivalent to the shake flask for the production of infectious lentiviral particles. Okay, in this section, I will be talking about some key steps for purifying lentiviruses. Obviously, with the lentivirus speed becomes the downstream lentivirus purification becomes an essence. Elongated time in the supernatant would definitely diminish your lentivirus infectivity. So once started, it's very imperative to have a streamlined protocol in place, which will enable you to keep the time to an absolute minimum from the start of the process to your final harvesting, followed by filtration and freezing down of the lentivirus. So what we have shown is a streamlined workflow that is transferable to any scale stir tank bioreactors. So in the step 8 of the process, which is a continuation from the stir tank reactors line, here we are showing that once that 48 to 55 hours is passed, benzenase and magnesium chloride is added to the bioreactor and incubated for a couple of hours at 37 degrees centigrade to allow for the digestion of any free nucleic acids in the bioreactor. And typically, this is where we take the samples for infection titer and total lentiviral particles using our HD 1080 infectivity assay and a P24 ELISA respectively. So here, it should be noted that we are working with an anti-CD19 CAR construct that has a V5 tag. And so in the HD 1080 assay, infected cells are being monitored through an anti-V5P conjugated antibody by flow cytometry. So following the benzenase treatment, the supernatant is clarified by passing it through the depth filter. And this filtration train may start from a 5 micron pre-filter down to a 0.8 or a 0.45 micron final filtration, which is transferred into a suitable bioprocessing container bank. Here, the additional samples tend to be taken to do a host cell protein monitoring through a HEC293 ELISA and the host cell DNA via qPCR or a digital droplet PCR. So now I'll move on to step 10, the chromatography process, which is a critical step in the lentiviral downstream processing. And here, quite typically, anion exchange resins, including our porous D50 CII-MD and CII-MQ resins have been used with great success to purify the lentiviral particle from other components of the clarified supernatant from step 9. In addition, there are other resins such as the heparin affinity resin that have also been used for LV purification. We've shown some references there, which you can take a look to see what exactly are the parameters that are used for this downstream processing. So the choice of the resins really depends upon your preferred process and steps and your lentivirus construct and the CAR construct as well. So in the next step, the final ultrafiltration or the diafiltration, diafiltration is carried out typically through a tangential flow filter, hollow fiber, and the diafiltration step of 6 to 8 diafiltration volume into a CTS optimum medium, followed by final filtration on 2 micron PES filter. Okay, and what we can see here, when we carried out an experiment, we actually ran our stir tank bioreactors with 2 different protocols. One that uses a standard PEI-based process and the other CTS LVMAX protocol. We found a couple of major differences between these 2 processes. In this experiment, the PEI transfection was carried out at approximately 2 million cells per mil, where LVMAX transfection was carried out at 4 million cells per mil. So as you can see from the table, the CTS LVMAX, through the use of enhancer in the supplement, is better able to handle high cell densities and therefore coordinately higher lentivirus final titers. So through the robust downstream process that I just described previously, the host cell protein, even though it is shown to be somewhat higher early on in the clarified culture in the LV-Max system, that is not unexpected given the double cell density of the system at the time of transfection. But what is clearly demonstrated here is through the downstream process in the AEX anion exchange step, which is clearly able to take care of the host cell protein and reduce it to levels slightly lower than the PEI-based process. So as you can see there, there is no issues in handling the additional host cell protein through the anion exchange regime and purification process that I mentioned in the previous slide. So this concentration of the host cell protein and the host cell DNA follows the recommended guidelines that Mike just shared in his previous session. So on the right-hand side, you can see that the final titer of the lentivirus that was filtered using the 0.2 micron filter, and we had about a 6-fold higher titer using the CTS LV-Max system compared to the PEI-based process, and thus it's kind of evident that we had about a 10% higher final lentivirus recovery for the CTS LV-Max compared to the PEI process. And lastly, to ensure the infectivity of our CD19 CAR lentivirus, we activated the PBMCs for 72 hours with the anti-CD3, CD28 dynabeads, and we transduced these isolated and activated T cells with our CTS LV-Max lentivirus, and we typically culture them for 5 days, and then we assess the anti-CD19 CAR T cells by flow cytometry. So the bottom left, you can see the data for the T cells that are transduced at various MOIs in a T cell medium with or without your polybrine reagent. We found that the MOI of 10 with a polybrine reagent generated more than 40% CAR positive expression. On the bottom right, we transduced T cells isolated from 3 independent donors, which were transduced with our CTS LV-Max lentivirus at a MOI of 10 with the polybrine, and in the flow cytometry analysis, you can see that the CAR T expressed cells located using anti-B5 antibodies, and the negative control, the non-infected T cells shown to have a very clean population with no cells showing positive B5 tag, while the LV-Max system shows a 40%, more than 40% of the cells are positive for CD19 CAR construct. On the bottom right, when you look at the T cells transduction efficiency, you can see that again that more than 40% transduced cells expressing the CAR construct are stable for over a week, as indicated by the average CAR expression of the 3 donors. The CAR T cells at days 3, 5, and 7 post-transduction. Okay, so in summary, I want to highlight that the CTS LV-Max system provides a streamlined solution for production of high-titer lentiviral vectors, and has an excellent scalability from multi-well to stir tank bioreactors. And as I've shown, a multifold improvement in your final viral titers compared to your PEI-based process. And the downstream processes are very streamlined, which is able to reduce your whole cell protein DNA. And the resultant purified lentivirus from the CTS LV-Max is shown to transduce human T cells in vitro with an anti-CD19 CAR construct. Okay, moving on. Next, I would like to talk about our next innovative solution, which is the porous capture select AAV X resin, a platform for AAV purification for different serotypes of AAVs. So as you know that the recombinant adeno-associated virus-based vectors have reported excellent clinical outcomes, and the AAV subclasses have shown greater potential for different gene delivery applications due to its impressive advantages as listed in the slide, and it's become a vector of choice for many therapies. Although excellent clinical outcomes have been reported, the point to be noted here is the current potential of these viral vectors in the clinic is significantly limited due to the manufacturing challenges, including the absence of an efficient and a scalable platform purification process. So the purification methods for AAV are often being developed for each new product in each isolated group, creating a large portfolio of methods which lacks a standardization. You can see that the purification process usually occurs at least in 5 major phases. The harvest of the producer cells, followed by the cell lysis, followed by a cellular viral nucleic acid removal, and particle separation by chromatography, and the concentration formulation. So the many steps in the process lead to high loss product, which can lead up to overall product yield of only 20%, as shown in the graph at the bottom right. So as reducing the amount of steps and adopting a scalable purification strategy will be critical in improving your productivity. So before explaining in more detail on the AAV affinity resins, I would first like to explain our affinity technology platform called Capture Select. So the Capture Select technology is based on the variable domain of camelid heavy chain only antibodies, which as a single antibody domain providing a full functionality in antigen specific replication and high affinity binding. So the heavy chain only antibody are the smallest antigen binding fragments, leading to a unique affinity for the target molecule. And because of this compact structure, these domains are very robust and can withstand the various conditions that's typically run in chromatography. So let's move on to the affinity solutions for viral vectors. I previously explained that the Capture Select ligands are immobilized to a chromatographic support. So for the AAV affinity resins, the AAV809 and the AAVX ligands are basically immunomobilized on the high-performing porous base, which is very suitable for purifying large molecules. So, for example, the capacities obtained for AAVX are greater than 10 to the power 14. You have to keep in mind that this is dependent on the serotype. So we have performed capacity studies with a collaborator in academia, through customer feedback from various evaluations, we're able to confirm this high capacity and specificity of these resins. So this slide shows the binding of the AAVX resin to various AAV serotypes. So the data was kindly provided to us by the Massachusetts Eye and Ear Hospital in Boston. Although a simple static behind binding experiment, it clearly shows that binding of AAVX to a larger variety of serotypes and recombinant vectors. So as a matter of fact, to date, the AAVX ligand has shown affinity towards basically all the serotypes, as you can see from the figure, which is tested by our customers. So the next question is, what about the scaling up? So you can see the yield and the post-purification at various scales. From here, you can see that the porous Capture Select AAVX resins shows comparable performance at various scales across different serotypes tested. So to conclude, this data shows that the porous Capture Select AAVX affinity resin binds to a broad range of serotypes, and it's very flexible and it's applicable as a platform resin for AAV purification without the need for optimization for different serotypes. Okay, so before moving on to the next section, I want to spend a few minutes to talk about our new and exciting prototype of a complete animal origin-free, a scalable, suspension-based, helper-free AAV production system that is due for launch in Q1 of 2021. So if you can see that the components of the AAV production system have been developed to work synergistically with each other to produce an enhanced titer of the AAV. So currently, the beta testing of this AAV production system is ongoing globally. So if you're interested to evaluate this media for your AAV production, please reach out to us. Okay, so this is the last section of my talk. So I want to talk about our non-viral gene delivery systems, the electroporation part. So on the ex vivo based gene modified cell therapy, lentiviral or retroviral transduction is safe for now. But however, our AAV as a donor DNA delivery system, along with the CRISPR-Cas9 RNP complex for gene knockout delivered via electroporation, is looked as a safer method because of the AAV virus's inherent properties, such as the low immunogenicity, ability to infect both dividing and non-dividing cells, and non-integrating as well. So the non-viral based methods present certain advantage over the viral based methods with simple large-scale plasmid production and a very low host immunogenicity. And that this is the specific reason that it is a preferred and a safest gene modification tool that is currently being explored in many ongoing clinical trials. So in this case, a naked DNA template encoding your gene of interest is transfected or electroporated along with the CRISPR-Cas9 RNP complex or a TALEN for gene knockout in order to achieve a typical lentiviral-based 20 to 40% of CAR expression. And to the point to be noted here is the non-viral-based methods require a significant protocol and reagent optimization. So having the best tool is highly important and critical to achieve your high editing efficiency. So currently we are the only company to offer a complete suite of gene editing solutions that include both the TALENs and the CRISPR-Cas9 nucleases. And we basically do provide an end-to-end service to support every step of your genome editing workflow. More specifically, the genome editing solutions that we provide are paired with optimal cell culture reagents, delivery methods, and analysis tools based on your application. So the genome editing is really simplified into two simple mechanisms, but I won't go through in detail. So the two methods are the gene knockout. Basically, there's a double-stranded break, and then the cell's own repair mechanism repairs through a non-homologous end joining, causing indels, leading to a frame shift or premature stop. And this is what we call the gene knockout. And the second mechanism is a more specific homology-directed repair, where a donor gene is provided, which is knocked into the region, which is typically done with a donor DNA insertion fragment. Okay, on the gene knockout, on the gene knock-in or the homology-directed repair, which uses the donor-based molecule method, which is shown on the right and left arm with the blue insertion sequence. And when you have the donor molecule present, you can have a specific change to the genome incorporated, which can be the blue box that is incorporated into the genome. So this mechanism is typically used to add reporter genes, create fusion proteins, disrupt any functional domains, or we could either mimic or correct your SNP changes that will change the respective protein function in order to study those disease models. So as you can see on the right, there are many different donor templates can be used to, which depends on either the function or the size of the insert desired, such as the single or a double-stranded DNA, or a plasmid DNA, or viral vectors such as the AAV. Okay, in this slide, I want to highlight the different types of double-stranded breaks that these nucleases produce. So TALENs, which is shown on the left, typically produce a double-stranded break with a 4 base pair overhang, while the CRISPR-Cas9 produces a blunt end break. So because of this 4 base pair overhang, we find that TALENs have very high and specific knock-in efficiency compared to your CRISPR-Cas9 system. In addition, the CRISPR-Cas9's restrictive access to certain areas of the genome due to the PAM sequence and availability, SNP changes at these sites are very challenging, while the TALENs have unlimited ability to target any sequence because of the direct binding capacity, and they can access those sites with ease. On the other hand, because of the blunt end break, CRISPR-Cas9 produces much precise and high efficiency knockouts compared to the TALs. Okay, in this slide, on the left, you can see the comparison of the knock-in efficiency using the TALENs for single SNP changes in 3 different genes compared to your TALEN and the CRISPR. Based on the data, it's evident and consistent that the TALENs have a very high HDR percentage compared to the CRISPR-Cas9 nuclease, as I mentioned before. So we provide a ready-to-use transfect TAL mRNA for genome editing. So for this service, we developed a robust algorithm that allows us to generate the most optimal the TAL effector designs, followed by in vitro transcription and RNA purification. Okay, so moving on to a CRISPR-Cas9 product called the TrueCut Cas9 V2, which was recently used in a landmark phase 1 CAR-T clinical trial to do a knockout endogenous TCR and a PD-1 in T cells. So more importantly, what they have shown in their data is that the edited T cells are engrafted in all the 3 of the patients are at stable levels for at least like a 9-month period, and no clinical toxicities were associated with the engineered T cells. So the persistence of the T cells were also seen to be more durable in these clinical trials compared to the previously infused T cells, where they did not knock out the endogenous TCR. So to further facilitate clinical trials, the question is how we can provide the TrueCut Cas9 in a regulatory compliant manner. So the answer is our new Gibco CTS TrueCut Cas9 protein to transition from the bench to clinical research. So what we provide here is extensive safety and the traceability documentation, and the manufacturing process is entirely aseptic. So the CTS prototype version is available now. If you're interested, you can have a beta testing program, and the full version will be available in 2021. So before I end this session, I want to highlight the benchmarking of the Gibco CTS TrueCut Cas9 prototype with our existing TrueCut Cas9 version 2 protein, and we found out that the knockout efficiency is more than 90% in human primary T cell populations, electroporated with our neon electroporator device. So on the left, on the flow cytometry analysis, you can see that the mock negative control data is clean, while the TrueCut Cas9 with the TrueGuide modified guide RNA against the track locus generated almost 93.9% functional knockout. On the right, a different concentration of the CTS was evaluated, and you can see that there's no difference between the different concentrations that we tested. So at the last, I want to end this talk by highlighting our electroporation system, where we successfully used to deliver genes and gene editing capacity from R&D and manufacturing scale. So you can see that the small volume, the neon electroporator system with this unique tip design provides an easier optimization and high viability. And we have shown with the data that neon can easily introduce 2 sets of Cas9 ribonucleoproteins and a long linear DNA to achieve double knockouts. So with the help of a newly developed gene editing buffer, we showed more than 30% knock-in efficiency and 95% of knockout efficiency in track locus. So now we are seamlessly transferring our neon's capability to a new large volume electroporation system, which is under development. This flow system automation system can accommodate 1 ml single shot or a 5 to 25 ml flow through reactions. So we already have data showing that gene editing and DNA delivery efficiency is equal or better as compared to neon. So the reagents and consumables are available with the GMP grade. So to conclude, the LVMAX system, I talked about the LVMAX system, which can solve your LV production challenge upstream and downstream. And the porous capture select resin serves as your platform technology. And then there's a broad range of non-viral gene editing technologies, such as the TrueCut Cas9 and the Talents. So thanks everyone for your time.
Thank you, Prem, for the presentation. We're now ready to take your questions. Please feel free to send your questions through the Q&A box, which is indicated by a question mark icon near the bottom of the screen. You can also send your questions if you don't manage to get them addressed during this session at the poll that we will open shortly after this. So we have an open-ended question at the end where you can actually put in your questions and any requests that you may have. So let us have a look at the Q&A box to see what questions that has come up. So a question from Michael: what is the current state of the art for LVD concentration or purification?
So the current state of the art for a lentivirus concentration or purification, I do not understand the question clearly. So based on our lenti LVMAX system, the concentration of the virus that can be produced can be like 10 to the power 8 to the 10 to the power 9 after your concentration process. So the concentration specifically can be done using your anion exchange resins that I mentioned, which would remove your contaminants such as the host cell proteins and the host cell DNA. And the purification process can be further enhanced using the ultrafiltration or the diafiltration process.
Thank you, Prem. And a question from Chu Wei. Are the electroporation systems 21 CFR compliant?
That's a very good question. So I just mentioned about the large-scale electroporator system that is that we are developing. So this is will be connected to OPC UA, so you can connect to your third-party 21 CFR device. So to answer your question, yes. So the new large-scale electroporator system will be OPC UA enabled.
Thank you, Prem. And I see a third question coming up. Do you have a suspension-based system for AAV? Our lab is shifting from adherent cultures.
Yes, so the slide that I mentioned, there's a helper-free AAV production system that I mentioned. So there's a beta testing ongoing, and we have seen impressive results from all our collaborators for AAV production. So basically the yield, what we observe is on a very high titer range of 10 to the power 13 to 10 to the power 14. So it's a suspension-based system, again, a helper-free AAV production platform. And as I've showed the slide before, it's a very simplified process where it starts with the high density cells and then animal origin free media coupled with your buffers such as the transfection reagent and the complexation buffers followed by a lysis reagent. So everything is given as a one solution for you to produce your AAV in a high titer.
Thank you, Prem. All right, I think these are all the questions that we have. Let me just check through. All right, so I don't see any other questions coming up. If you have any other questions, please feel free to type it in the Q&A box, and we will have them addressed by our panelists during the next presentation. So we have a poll coming up next. So I'm going to pass this over to Sonia to open the poll. Over to you, Sonia.
Hi, Lydia. Hi, everyone. Can you hear me?
Yes.
All right, great. So right now it's poll time. So I will need everyone to look towards the right side of your screen where our poll will be coming up very shortly. So as earlier mentioned by Lydia, there will be 4 questions and there will be entirely 4 polls for this 2-day conference. So if you participate in more polls, you will have higher chances of winning our Logitech headset as well as a very exciting UV sanitizing box. So for this Logitech headset, it's worth about 90 Sing dollars. So it's pretty good if you need to deliver a lot of presentations at virtual conferences or conducting scientific webinars. All right, so let's start with the poll. Today we have a total of 4 questions. So if you have any other questions from Dr. Prim's talk, you can include your answers. So first question, what viral vector are you currently using? And if you are interested in any of the products, and question 2. And of course, if you have further questions, which you might like to arrange a follow-up meeting with us via a quotation or meeting with a Thermo Fisher sales rep, please let us know in question 3 as well. And for Southeast Asia customers, we will also follow up with our distributors to arrange a meeting with you accordingly. All right. And if you have any other questions regarding products which are not mentioned in today's session, feel free to let us know in question 4 as well. All right, we also have quite a couple of researchers who asked for a certificate of participation. So if you really need a copy, please let us know in question 4. Okay, so we have around 3 minutes left. So feel free to participate in the poll, take your time. And if you have any other questions which that came up to you, you can actually type down in the Q&A box after this poll. Dr. Prim will be here till the end of today's session to answer your question. All right, for those who have completed the poll, you may go for a short coffee or bio break. And next up, we'll have Dr. Ryan Lin to share about the next exciting topic, which is about cell and gene therapy identity, priority, and safety testing. Thank you for joining us at Gibco Galaxy Tour 2 since 9 a.m. Singapore and Taipei time this morning. Please stay with us as we still have one more talk going on. All right, we have around 2 minutes left for those who are finishing up. All right, we still have a couple who have not started on the poll. So if you guys are not able to see the poll clearly, you can expand the tab. So you should be able to see this arrow tab beside the word polling, and you can expand it, and then you can see the questions. So there should be a total of 4 questions that you will need to complete. And as mentioned, if you complete all the polls, you will have a higher chance of winning our UV sanitizing box, as well as the Logitech headset, which we'll be announcing on 14 October. And for those who have completed, thank you very much. Please stay on for our next session. And of course, for question 4, if you have any other feedback or any other topics that are related to cell and gene therapy field, do let us know. We can come out with when we curate more content next time for our next Gibco Galaxy event, we can consider your feedback as well. So please let us know your feedback. We would love to hear them. Share with us. All right, thanks for staying on with us at Gibco Galaxy Tour 2 this morning since 9 a.m. And right now, we will be closing the poll, and I'll pass the time back to media. Over to you.
Hi, Sonia. Thank you very much, Prem, and thank you very much, Sonia. So I'll now like to introduce our third and last speaker for the day, Dr. Ryan Lim. Dr. Lim is our regional field application scientist and with a focus on cell and gene therapy product characterization, product quality, and lot release testing. He will be providing an overview on the current trends, technologies, and methods available to ensure that cell and gene therapy products are safe for release. Again, if you have a question for our speaker, please send it through the Q&A box. And now, let us give a warm welcome to Ryan. Ryan, over to you. Sorry, Ryan, I think we're not able to hear you.
Okay, can you hear me now?
Yes, you're good now. Thank you.
Yeah, apparently I was on mute, sorry. Okay, so hi everyone, hope you all can hear me well now. So my name is Ryan, as Lydia shared, and I'm supporting basically the immunoassay and immunotherapy portfolio. Right, so today we heard great talks about, selecting the right starting material and, our next generation solution to accelerate your gene therapy by Prem. Now, if you notice, the underlying theme that we have going on here is basically how to fast track your work. So this is the last segment for the day, and I'm going to do the same thing, just that, but we're going to focus on characterization. So how can we help you speed up your work when we look at characterization? So firstly, what is characterization? Why do I need to do this? When do I need to do this? How do I do that? So hopefully, by the end of my session, you guys will know all the answers to these questions, right? You guys will be the experts here. Right. So first up, very simply put, characterizations is a critical quality standards that are proposed and justified by the manufacturer and approved by your regulatory authorities as conditions to move your therapy forward. So it establishes the set of criteria which a drug substance, a drug product, or material should conform to be considered acceptable for its intended use. So it normally consists of a list of tests, references to analytical procedures, and appropriate acceptance criteria. So the release specifications are just one part of a total control strategy designed to ensure that your product quality and consistency. So hence, this is something that needs to be done. And I stress again, this is something that you need to do if you intend to move your work into a clinical trial or if you are going to the manufacturing phase. So this is something that's unavoidable. If you're going into clinics, if you're giving it to patients, if you want to move it forward, this is what you need to do, right? So given that we say this is what is needed, what exactly do we need? So from the US FDA guidelines, which was again, as Mike shared, recently updated in January 2020, we can actually separate them into 3 arbitrary categories, namely identity and purity, potency, and safety. So let's take a brief look at what each of them are. So firstly, identity and purity. So basically, you need to demonstrate that your cell type is what you say they are. You need to perform specific assays to ensure the identity and purity of the final product, as well as what kind of variations would be acceptable to you. You know it's not possible to get 100%. So what is the ratio that you are comfortable with? Next up is potency. Again, very simply put, you need to provide substantial evidence that your product has clinical efficacy. Now, there is no one single assay that can demonstrate sufficient clinical efficacy, and you will likely have to measure multiple parameters. Now, most importantly here, if you do not provide what your regulators deem as sufficient, your pivot trials can be put on hold. So that's what we call a clinical hold. You really can't move forward. So imagine all the work you put forward, all the work you have done stops right here. Now, as we shared in our first talk, so lastly, we want to talk about safety, right? So testing is needed basically to evaluate that your final product is safe for use and it does not have any advantageous agents. So even if your product, it has clinical efficacy, if it's not safe, it doesn't matter. So as we shared in our first talk by Casey, careful consideration on your starting materials, your ancillary reagents should be done early on, as they might impact the safety of your final product. Now, as rapid progress is made in cellular therapeutics, characterization remains one of the key challenges in cell product manufacturing. It often requires a significant number of cells. Therefore, today's needs are in minimalizing your sample size, while using predictive assays that are scalable, easily standardized to establish fit for intended purposes. Now, the real goal of these assays in the future will be to continuously monitor for better process control and real-time release. So we are talking about easy-to-use assays with support for implementation and validation of the assays. So these are all its critical requirements. So as such, Thermo Fisher has been focusing on our next generation technologies to address these issues. As Prem has shared some of them with the upstream, I will be now looking at what we always see as the downstream of the workflow. Right, so this is a typical cell therapy workflow. So as you notice, characterization and lot release are normally put at the end. However, the truth of the matter is, it should be done at each stage of the process. Think about this. Would you prefer to know if part of your process has some concerns early on, or would you rather only find out right at the last step? After you've invested all your time and money, surely you will pick the former. right? If you know that your gene engineering or your cell expansion is having issues, you would definitely stop there. You won't keep proceeding. So this is where these tests will come in. So as I said, I'll be covering on 3 separate categories. So we shall start with the first one. Let's look at identity and purity. So when we talk about cell therapy, there are many different cell types that researchers are working on. As such, different criteria are needed to sufficiently identify the cell type. So what kind of cells you're using? Are they the same cells? Are they still functioning properly? Now let's use MSCs as an example. So on the left, these are the minimal requirements as set about by the ISCT to define what makes an MSC. And because we know this, we at Thermo Fisher here, have developed assays to help you measure these parameters, such as our tri-lineage differentiation kits, as well as our flow assays, our flow cytometry assays, that can measure the presence and absence of your surface expression markers. Now, in addition, we have also designed assays that can work simultaneously together. Remember, one of the concerns we shared was, with sample types, with sample size and all that, and time. So this is something that we combine them together to save you on your samples and time. For example, our ClickIt-edu proliferation assay can be coupled with our antibody phenotyping panels for use of flow cytometry. Now, given the versatility of flow-based assays, having a good instrument is just as important, as this will allow you to speed up your work. So as such, we have our own flow cytometry instrument, our Attune NXT, which I'm very certain all of you have heard before, right? So additionally, we have this now as a 21 CFR compliant software, which, as you know, is necessary for all your audit trails. Now, I won't go too deep into this instrument as it would be a seminar in itself. So rather, I just want to share with you that this instrument has the ability to, in a sense, protect your samples, your very, very precious samples from losses. It has a simplified sample prep. It's able to do high throughput, and it's actually suitable for full automation. So now, if you're curious to know more about this fantastic instrument, again, please reach out to your accounts managers. They can help you explain a bit more, or they can link them up to you. Now, remember how I mentioned that characterization assays should be done at each step? Well, in addition to just identifying cell type, characterization at the early stages can also give you insights on several critical questions during your process development. Now, we can't talk about cell therapy without talking about CAR-T therapy. So hence, using that as an example, some critical questions that you will want to ask would be, are my starting samples ideal for cell manufacturing? Or, what is the purity of the cells isolated and expanded? Additionally, questions like, are my cells expressing my gene of interest? Or are my cells proliferating effectively? Would elucidate if you should carry on, or if something is not optimized and you should not waste time proceeding further. So again, we have our cell trace assays to help you see if your cells are proliferating properly, as well as our prime flow assays that allows you to measure your gene expression at a single cell resolution. You can actually know if your individual cells are functioning properly. Now, one other very exciting innovation that we are expanding on is our PureCone assays, which are used to identify various cell types using qPCR. So these are the recent launches that we have. So let me explain a bit further, why we decided to do it. Now, first of all, the standard method is using flow cytometry. And flow cytometry is a workhorse method, and it's used by almost all cell therapy developers, just due to its versatility and how good it is. However, it is not without its challenges, and especially as you move to establishing its use in the GMP manufacturing environment. So there are issues with logistics, with costs, with variability, and training is necessary to mitigate all these operator variability and subjective analysis due to, say, different gating strategies. So as such, our approach in Thermo Fisher is to basically continue to support flow-based methods with qualified antibody panels. We provide you with the best reagents as well as to support training to help reduce all these issues. Now, in addition to this, we also explore, alternative methods that are quantitative, easy to standardize, and ready to be implemented in a GMP manufacturing environment. So with that, I'd like to introduce to you our pure quant assays. So we recently launched our pure quant assays, a PCR-based method, which offer a more standardized and reproducible data compared to flow cytometry. And this is to measure immune cell type in a heterogeneous sample. So this is not new technology. Rather, a company, Apiontis, has been using this technology for over a decade, and it is well accepted by the industry for patient monitoring. Now, this assay uses a unique demethylation site within a gene that definitively IDs a specific cell type. So when used in loci with cell type specific epigenetic patterns, this technique can be used to detect and quantify a wide range of different immune cells. So using this assay, subsets of T cells was identified and absence of contaminating cells such as B cells and monocytes were confirmed. Now, all our assays are qualified as per ICHQ2 R1 guidelines, making it relatively easy for further fit-for-purpose testing and implementation for process or product characterization. Again, if you wish to know more about this assay, please feel free to reach out to me, and we can schedule a separate discussion where I go through, exactly how this works and what are the steps and what are the pitfalls to look out for. Right, so that is identity. Now let's talk about potency. Or simply, is the therapy working the way it should be? Now, I think it is fairly obvious why we need to provide evidence for potency. But what is not so obvious is how should we go about doing it and what exactly is needed. Now, Thermo Fisher Scientific has developed multiple assays to test for potency. But today I'm just going to focus on two assays. Now, the first one, which Mike has briefly touched on, is our Invitrogen Cyclone LDH cytotoxicity assay. So if you have been with us earlier on, you would have seen this slide as well. And basically, what I want to share from here is that we all know what CAR-T is used for, right? Its main function is to kill cell. So as such, it is critical to demonstrate that your CAR-T product can do just that. So a direct assay to consider would be this cytotoxic assay, where our Invitrogen Cyclone LDH cytotoxicity assay is a colorimetric assay, and it provides a simple and reliable method for determining cellular toxicity. Is your cells dying? So LDH stands for lactate dehydrogenase. So this is actually a cytosolic enzyme present in almost all the cell types, and it is released into the cell culture media upon damage to the plasma membrane. So for our kit, it is a full kit. We provide you with all the reagents to accurately quantify and measure this extracellular LDH. So in this data, you can see the potency of 3 CAR-T cells, right? 3 donor CAR-T cells. They were determined using our Cyclone LDH assay. So the untransduced T cells, as you can see in the orange, which you don't see, except in the they are our control. So our in vitro cytotoxicity was detected with this. And you can see that CAR-T cells exhibit a higher LDH signal, indicating a higher percentage lysis than the untransduced control. So percentage lysis for all these donors were shown after a 16-hour cold culture with our CD19, our K562 cells. Now, the second assay, in addition to cytotoxicity studies to test for potency, we also test for cytokine secretions of these 3 donors. So cytokines were produced after an overnight stimulation with our CD19 expressing K562s were detected with our 34 plats, and signal intensity was reported as an expression level. So we can see here that our CAR-T cells produce higher cytokine signal than our untransduced control, a bit with differences in your expression levels of different markers because of the differences between the donor cells. Now, you might be thinking, why cytokines? And why are we testing so many of this? Well, the reasons researchers and companies are testing them is because many cellular activities are modulated by your cytokines. So looking at MSC as an example, you can see that its immunomodulatory potential has been attributed to its secretion of cytokines. Now, in fact, if we look at our current COVID-19 pandemic, in which there's a link between its severity and cytokine storms, you can see that several trials are actually being done using MSC as a therapy due to its immunomodulatory functions. And in fact, there have been some success as well. Hence, it is critical to measure, what are these cytokine expression levels. So that is what a lot of people are looking at. The cytokines that it secrete has the ability to affect other parameters as well. Furthermore, cytokines actually play a role in effector function as well. So take, for example, in your NK cell, which different classes of cytokine plays a different role in effector function. And the absence of a class would significantly affect its overall efficacy. So you can see it as the ability to release cytotoxic granules, cytokines, or the death ligand death receptor mediated apoptosis. So imagine if one of these was not manufactured properly, or there's an issue, its overall efficacy will be down. You are still killing the cells, yes, maybe through the release of cytotoxic granules, but its efficacy is greatly reduced if you do not have the function of the death ligand death receptor mediated apoptosis, for example. So this is why it is actually critical to see cytokine activity, and rather a lot of cytokine activity as well. It's not just a single target. Now, likewise with CAR-T products, cytokines play a very critical role, and it is to the point that a commercialized CAR-T product states the type of cytokines that they tested for in their product insert. So as such, I hope I demonstrated to you, that cytokines are very, very important. And this is why so many people are looking at them. And that the different cell types that you're looking at, whether it be MSCs, NK, CAR-Ts, the kind of cytokines that you need to measure is going to be different as well. Now, if you understand that, then you would actually realize your next problem. And that is, there really is a lot of cytokines to test for. So how do you test for so many? So with that in mind, I would like to introduce you to our next generation immunoassay, our Procardaplex. So as I mentioned previously, the challenge with testing cytokines is the complexity that comes with so many targets. What do you test for? What is the cost? These are factors that comes in. Secondly, think about how long it would take for you to test so many cytokines. If you use a standard ELISA, it's important, you have to do it. But how long will it take for you to do it? Lastly, with so many plates, how accurate would your data be? How reproducible will it be? How do you compare the data across multiple plates? All your plate variation, your lot-to-lot variation, so on and so forth. So Thermo Fisher understood this, and that's why we also came out with our ability to answer all these challenges, and that is with our Procardaplex. So our Procardaplex assay actually functions just like a sandwich ELISA, but the difference is that you can measure between 50 to 80 targets in the same well. So that means you only actually require between 25 to 50 microliters of sample for all these targets. And you can run them all in one day at a fraction of the cost if you were to run them all on multiple ELISAs. Right. So, it does sound too good to be true. So let me explain to you very briefly, how does it work? Well, firstly, we utilize this Luminex XMAP technology. So these are basically magnetic microspheres that are dyed with two different dyes at a particular bead to dye ratio that gives it a very unique signature. So this gives it its unique ID. Now, using these unique beads, what we did was we add our antibodies onto them. So hence now each antibody has a unique identifier. Now, if you see on the right, with a standard ELISA, the limitation is that you can only tag one antibody to the bottom of the plate. So like this example I showed here, if you're looking at 4 targets, you will need 4 plates. If you're looking at 50 targets, you will need 50 plates. Now, this is not so for the Procardioplex, as you can add multiple targets into a single well. This is because each target is uniquely tagged with a bead. Thus, all you need to do is basically add them in, run the standard protocol that we have, and then measure it with the Luminex instrument. So as such, you can measure all your cytokines in the same well. Now, and this will save your sample. It will definitely save you time, money, as well as increasing your accuracy. So we have an extensive library. So you can purchase your Procardioplex as ready-made panels, just like the 34-plex that I showed earlier and that Mike shared as well, because these are targets that our scientists have already curated for you. Or you can buy them as individual targets as well. Now, most interestingly, we have the ability to build custom panels for you. Okay, I know custom is a really scary word to most researchers, but this is not that kind of custom. So what we mean is this. Now, unlike our other competitors that only sells you panels, fixed panels, right? And you can't even remove the panels or remove the targets if you're not interested in them, Thermo Fisher allows you to purchase them a la carte. Basically, you can pick and choose your targets that you want, or you can just remove targets from our panels, and then we will just charge you for the targets that you want. We won't charge you extra for that. Now, this process takes roughly about 5 working days, as we need to also QC this new kit for you. So it's a very fast turnaround. The cost price is similar to the kits, so you're not paying a premium for that as well, right? So this is something that a lot of our users employ. So they start to screen with a larger panel first, right? Based on their genes of interest, they start to screen on a larger panel of cytokines, and then they identify their targets of interest, following which they will hone down to a smaller list of targets, and then they'll get us to customize that, thereby reducing their costs, right? So this is something that a lot of you can use as well. Now, I do know there's always concerns, with cross-reactivity, but please be assured, that we have thoroughly tested our targets and ensured that our targets that we put together do not have cross-reactivity, and that our targets are also scalable. So even when you go down to smaller panels, because we guarantee that we use the same antibodies, the data is consistent. And as I mentioned, we use our own antibodies, we make our own antibodies, we also make our own ELISAs. So, 80% of our targets and ELISAs are mappable. So therefore, this allows you to, hone down even to an ELISA if you need to. So you can start with a Procarda, you can hone it down to the targets you're interested in, and then suddenly look at ELISAs if you wish. So as I mentioned, a Luminex instrument is necessary for the Procardioplex to be used. However, in addition to running protein quantification assays, your Luminex instrument can also be used to measure mRNA and DNA as well. So our QuantiGene assay, that's what we name it, utilizes the same instrument and allows you to quantify up to 80 genes, direct from your sample. You don't need to do cDNA conversion, right? You don't need to, purify, cDNA, convert, nanodrop, clean it up, everything. You don't need to do it direct from sample. Take your sample, lyse it, and then run it. And because we count the exact number of transcripts, we are not amplifying the number of transcripts, just the signal, you get a more accurate count. So if you're looking at multiple genes of interest, in your product, this again might be something for you. So once more, if you want to know more about this, just reach out to me, reach out to us, and the team can definitely help you with this. Right. So we're going to reach our last section, right? We talk about identity and purity, how is that important, and why we need to know, our cells are the cells we want. We also look at potency as well. See, all the cytokine testing. So now we look next at safety. So just like the previous two, this is also something that is required by your regulators. So this is a must do. And then basically, is your therapy safe and free from any advantageous agents or unwanted agents as well? Right, so I stress again, as with all the previous segments, this is something that you need to do. Now, again, there are multiple parameters to investigate when we talk about safety. However, two very important parameters that are again required to be tested for by the US FDA is microplasma and endotoxin. So early on, Mike gave a very good, talk about microseq, right? Our assay to measure microplasma. So I won't be repeating that, but rather I'll be more focused on our endotoxin detection assay. So our Pierce chromogenic endotoxin quant kit. Now this is a very highly sensitive endpoint assay. So this accurately measures and detects endotoxins in a protein, peptide, nucleic acid, or whatever sample that you want. And it uses an amoebocyte lysate assay. So this kit enables detection within 2 linear sensitivity range. So we're looking at 0.01 to 0.1 EU per mil and 0.1 to 10 EU per mil. So yes, 2 standard terms. So additionally, this assay is specific with no interference from your glucagon. And this can be done in about 20 minutes. Now, these endotoxin levels in the samples are accurately determined using our endotoxin standards of known concentrations. So these are derived from our equalized strain, the 0111B4. So like I mentioned, this is an endpoint chromogenic assay. So because of all the speed and all its usability, you just need to use a standard plate reader at a 405 wavelength. So that's all you need, just a plate reader and this kit. Everything else is already provided. Now, if you don't have a plate reader or are looking for a new plate reader, we have our new multi-scan Sky High microplate spectrophotometer, as well as our VeroScan Lux multimodal microplate readers. And these readers are actually optimized to our assays and to our plates already. So again, if you want to know more about them, please reach out to us. We have other scientists that are very proficient in them. They can help you out with demos, with explaining every step of the way and guiding you during your process development as well. So yeah, reach out to us if you want to know more about, this assay and this instrument as well. Now, lastly, I just want to share with you this protocol. So this map, as you can see here, right, is basically submitted by Novartis to their regulators for their commercialized CAR-T product. So one thing to note is that in addition to all these product release testing that they did, as you can see, they did all this release testing showing that everything is working fine. They also did in-process testing during their manufacturing process to ensure that everything is working as expected. So everything is working fine, from start to finish. This is what they did. They did a lot of in-process testing. And this is critical, should anything not be optimized, you can just stop it right there and then, just stop. Then you can reevaluate, where do you go wrong? Is it because you didn't look at your raw materials? Is there an ancillary reagent inside there? What is happening that is causing something to go wrong? And you can just, stop there and restart rather than, finishing all to the end. And then from your final release testing, realize, hey, I can't use this. So this is what in-process testing is critical. It really, really helps you to save time, and it really does speed up your work in the long run. So as such, we have come to the end of my session, and I do hope that you have found it useful. So I just have, 3, key takeaway messages for you to sum up my session, and just for you guys to remember this. And firstly is to establish your key characterization parameters early. So as mentioned by our previous speaker, if you intend for this to reach your patient, to reach manufacturing, you need to use the right starting materials. Right starting reagents, assays, and this is to avoid all your future barriers to success. It is really bad if you did everything all the way to the end and then realize that, because of your starting material is not right, you have to repeat everything before you can go back into the patient. So this is critical. Establish, what are the parameters that you really want to look at first. Now, secondly, is that cytokine expression is critical for multiple aspects. So given the myriad roles that cytokine plays, there is a need to measure and establish cytokine signatures for potency assessment in your manufacturing process. So a lot of people are already looking at this, and this is really something that, everyone is looking out for, as it gives a really good marker, whether your thing is going to work fine or not. So this is something that you should be looking at as well. Now, lastly, is to maintain consistency with an efficient workflow. So this is to basically help you achieve, higher throughput, that is reproducible as you scale up and scale out. So, today we have shared a few technologies that can help you do that. From what Prem shared, from what Casey, from what Mike shared, and what I shared as well. Basically, these are technologies that is to help you, speed up your work. So if you wish to know more about it, again, please feel free to reach out to us. We can discuss your work together with you, and we can see how we can help you implement it with all our new technologies. So we just shared a few today, but tomorrow we have an even more exciting lineup for you with a new innovation that we believe can truly change the way that you process your cells, right? So I won't give too much about it away and leave it for tomorrow's speaker. But suffice to say, it's not something that you want to miss, right? Because it will really accelerate your work. So stay tuned for tomorrow's talk. And with that, I just want to end with saying that, we at Thermo Fisher, we are really here to help you. We have many tools, we have many resources available to assist you with your work. So if you have any queries, please feel free to reach out to us, to our team of scientists and experts, in the various aspects of this field. And we'll be glad to assist you. Right, with that, thank you very much for your kind attention. Back to you, Lydia.
Thank you, Ryan, for the presentation. So there are a couple of questions that have come up on the chat. I'm just going to read them off. The first question, do we monitor the potency of the cells post-transfusion in the patients?
Sorry, come again.
Okay, sorry. Do we monitor the potency of the cells post-transfusion in the patients?
Post-transfusion, that's actually a very, very good question, and there are people that really are looking at it, but it's not really of the cells per se, but of the patient itself. So when you transplant back into your patient, right, you actually measure a before and then you measure an after. So that's something that people do as well to monitor, especially for things like CAR T therapy, where you're looking at cytokine storms and you're afraid of it. So this might give you a marker beforehand as well.
All right. And the next question, are these potency assays standardized currently for cell and gene therapy manufacturing?
So that's also a very, very good question. So the thing is this, I had a saying from a very wise person that says, cell and gene therapy, the field, right, is basically trying to fly a plane while building, right? So that's the thing. We are still in the midst of establishing, what is necessary, what is effective, what is good, what is standardized. So we honestly do not know. So as you can see from what CAR T released, right? When our first CAR T was released, which is Kymria, people were basically looking only at interferon gamma, so on and so forth. Now with Yescarta, the second commercialized product, we are looking at even more cytokines. And the third product that is still in trial testing, they're even looking at even more cytokines, so on and so forth. So the short answer to yours is no. And solely because there is no consensus to what is the most critical cytokines. So that's why we always recommend people to do a screen. To screen, what are all the cytokines that you're looking at? And then you pick out the key important cytokines which you believe is for function. And then, you start looking at them and establishing them as your set protocol. And that's where Thermo can help you out because we can customize that particular kit just for you. We will QC it, we give you a certificate of analysis, and that is not an additional cost. It is going to be the same price as if you buy an off-the-shelf panel.
Okay, thank you, Ryan. Another question on potency testing. At which phase of the CGT manufacturing do we carry these out?
Okay, so for CGT testing, I would say, okay, so what you need, as in the required by regulators, would be at your final lot release testing, right? You need to show that everything is working fine. If you are using as potency, you want to show that, yes, this is potent, and yes, it is consistent. So that means my first batch, my second batch, and all my subsequent batch, my process is consistent because my cytokine expression levels, they're going to be consistent. So you do need to show that at a potency step, at the last lot release step. However, we also suggest that you show it as your in-processing step within product development to see that everything is working fine as well, that there's nothing wrong with your manufacturing process, that is still consistent and robust at the starting end. So short answer, you definitely need it at the end, but we always recommend that you do it at multiple steps so that you know everything is working fine.
Thank you, Ryan. Question here about differences between Procata Plex and multiplex assays from other companies.
Okay, so the key difference between ours and all the other companies is that we use our own antibodies, right? So these are the Invitrogen, the e-bioscience antibodies that we use, that we have validated. So if you can read our Nature paper, we are the only company that does advanced verification as required by, or as requested by the antibody working groups. So this is something that we are very confident in, is with our antibodies. Now, the second thing that we can do is allow you to do this custom a la carte option, where you can pick and choose the targets that you want. So other companies, you can only buy fixed panels, and you can combine those panels together. But for us, if you don't want a target in our panel, you can remove it, and then we will give you just that fixed targets as a kit. So that's the difference within us.
Okay, thank you, Ryan. And then a question from Zui. If I integrate Procata Plex into my release panel, will I be informed of any critical reagent changes in the offered kit?
Yes, definitely. So if you are getting this, Procata Plex as a release panel, then we will have a separate conversation with you to ensure, you get a fixed lot. We'll give you master lots. Basically, we'll work with you deeper on all this as well. And if there is any critical reagent changes, which we do not foresee in the coming future, you will definitely be informed first as per our agreement. So this will be something that we work closer with you. And this is something that we always share with you as well.
Okay, thank you, Ryan. So I think those are the questions that we have. We are slightly ahead of time, but I don't see any other questions coming in. If you have a question that you have in mind and you may not be in time to type this out, you can always put this in the poll that will be coming up next. So over to Sonia now for the poll.
All right. Hi, Lydia. Can you hear me?
Yes, I can.
All right, great. Hi, good morning or good afternoon, everyone. Once again, so our poll is coming up very shortly. So please look towards the right side of your screen, and you may
Hi, a very good morning once again to everyone, and welcome to the second day of our 2020 Gibco Galaxy Tour. If you have joined us yesterday, welcome back. My name is Lydia Gan, and I'm one of your hosts for today. This morning, we'll be starting with a presentation on GMP compliance by our Senior Global Cell Culture Scientist, Mary Kay Bates, and our Regional Product Manager, James Tan. We will then take a short break of a few minutes before starting at 10:15 based on Singapore and Taipei time. With a key highlight of this two-day series, which is the unveiling of our new Gibco Rotier CTS counterflow centrifugation system, which is set to launch end of next week. Our regional cell and gene therapy manager, Jacqueline Ng, will be presenting this first in English, followed by our product manager, Jasmine Seng, who will be repeating this session in Mandarin at 11:30, again based on Singapore and Taipei time. So you can choose to attend either the first session in English at 10:15 or the second session in Mandarin at 11:30. All right, before I hand over the mic to Mary Kay and James, a few housekeeping items as usual. First, all of these presentations will be available on demand after the live session, and these are all accessible through the same Thermo Scientific link that you have used to register for this webinar. Next, if you have a question for our speaker, please feel free to send it through the Q&A box near the bottom of the screen. We have panelists on standby to take these questions, and we will also be addressing them verbally at the end of the session. And last, at the end of each of our presentations today, we will be inviting you to participate in a poll with us, during which you can share any questions and feedback you have for us. All poll participants will be eligible to win a Logitech wireless headset or a UV sanitizing box, and we have a total of 2 units to give away. Winners will be announced on our website after the event. And now, please welcome our presenters for the first session. Over to you, Mary Kay and James.
Thank you, Lydia. It's great to be here. I hope everyone is enjoying the event. What a great collection of presentations. And now I just have to advance my slides. Lydia mentioned the Q&A, so if you have questions, feel free to type your questions into the Q&A section at the bottom right of your screen. And as Lydia mentioned, there are a lot of chances to win some of these great prizes, the UV sanitizing box and the Logitech wireless headset. So that's pretty exciting, and I hope you'll enjoy it. I'm going to start out with the talk, and my colleague James will join us at the very end. So let's get started. The title of our presentation is Transitioning from Cell and Gene Therapy Research and Development to GMP compliance. Now, this is a really big topic, so I'm not going to get into the step-by-step detail. I don't think anybody can do that. I think that would require quite a few textbooks, and probably by the time those textbooks were published, they would be out of date. So this is what we're going to talk about today. We'll begin with a little introduction. What do we mean when we say regenerative medicine? That's a big term. So I'd like to start and make sure that everybody is starting with the same definitions from the same place when I go into a talk. So that's going to be the introduction. Then we'll review the global growth of regenerative medicine. Why is there so much excitement around this topic, and really what brings us all here today? And then the meat of the talk is part 3. What is GMP and why do we have it anyway? Then we'll go into some important ways that laboratory equipment can help you along your road to GMP compliance. And that leads us into, James will finish up with data logging and downloading. So, big picture, cell therapy has existed for decades. Blood donations, plasma donations, bone marrow, organ transplants, vaccines, something we're all thinking a lot about right now, biologics such as immune stimulators, cytokines, antibodies, and so on. These have all been around for decades. So cell therapy has existed for decades. Now, if we think about big picture regenerative medicine, I think the first thing people think about is stem cell therapy. But this is really only one small part of regenerative medicine. A big part of it is cell therapies not involving stem cells, so musculoskeletal cells, muscle cells, chondrocytes, tissue engineering, bone therapy, joint replacements. A big part of regenerative medicine and a big part of where the excitement is now is immunotherapy. I mentioned vaccines and biologics. It also includes natural killer cells. T-cell therapy is a big area of focus and growth right now, and especially one part of T-cell therapy, CAR T-cell therapy. Touching all of these areas is gene therapy, genetic engineering, primarily using viral engineering, but also non-viral types of gene therapy, including transposons, for example, or DNA or RNA carried by lipid nanoparticles, which is a couple of the potential vaccines for COVID-19. So that's just kind of an introduction of, again, big picture regenerative medicine. So let's look at the global growth of regenerative medicine. Before we do that, we have a polling question. So we want to know, in what type of institution do you work? And so in the Q&A section at the bottom of your screen, you can type in A, you're in an academic or government lab. B, you're in a small biotechnology company or a startup company. C, you're in a big pharmaceutical company or a large biotech company. Or D, a pharmaceutical company. Maybe you work for a contract manufacturing organization. Or F, none of these. And remember, when you enter our poll, you have a chance to win one of our fabulous prizes. Okay, so projected growth for regenerative medicine applications. This is new data just released actually a few days ago. And so the largest area of growth is actually in cell therapies, including immunotherapy, oncology applications, and musculoskeletal. I'm having problems saying that today. Actually, the smallest part of that market is stem cell therapies, and that's actually the smallest growth as well. So if we dig into some of the applications, I have another chance to try and say this, musculoskeletal disorders will remain the largest chunk of the applications, but oncology is really the fastest growing. So you can see across the board there, all of these have double-digit cumulative annual growth rates projected for the next 5 years. So, speaking of applications, here's another poll question. Which of these terms best describes your current application? And again, fill out your answers in the Q&A box. So A, musculoskeletal disease, cancer or oncology is number B. C is wound care or skin. Are you doing any skin research, burns, trauma applications, things like that? Dental care is number D. Eye disease is E, or F, none of these. There are many applications, so maybe your application doesn't fit into one of those big growth puzzles. So where are these regenerative medicine institutions? Where is all this growth occurring? And here's a map around the world. And as you can see, North America has probably the largest number of regenerative medicine institutions. This is according to the Alliance for Regenerative Medicine. But there's a lot of growth. If I just focus on Asia, where that number is 184, 1 year ago that number was 166. So actually there is a lot of growth just focusing on Asia. And Oceania, they've broken out into its own piece of the puzzle this time, and there's a lot of growth there. And actually that's number 22 is pretty well represented for kind of a small part of the world. Now, what types of institutions are making up all of these numbers? I think when we think about regenerative medicine, the first thing we think about is large pharmaceutical companies. But I want to point out those companies are only 8% of the total. The more traditional routes where a lot of the innovation is occurring are in nonprofit institutions and academic institutions with 23%. And then the largest part is smaller companies and startups, which a total of 64%, but those with fewer than 100 employees is 50% of the total. So I think maybe you joined this talk because you're making that transition, and maybe you work in one of those startup companies. We're glad to have you, and so let's continue. So with all this excitement, what is driving this growth? The biggest piece probably, depending on what your source of your statistics is just that we're seeing more and more disease across the world. As our global population grows, there's more cancer, there's more diabetes, for example, and so there's a greater need to address these diseases and disorders. Putting that aside, what's driving this growth is collaborations. Unprecedented collaborations, large companies like large pharmaceutical companies partnering with startups because that's where the innovation is occurring. So the large companies can offer infrastructure and technology and scale, whereas startups can offer the innovations, the new discoveries. Also, academics and government institutions are working more with industry. That's not something that happened a lot in the previous century. Funding. Investors are seeing the excitement and the growth happening in regenerative medicine, and so there's a lot more investment. There's also mergers and acquisitions where a large pharmaceutical company or a contract manufacturing organization might buy a smaller startup. And licensing technologies. So if you have a great discovery and you want to continue to develop that yourself, you may at the same time license that technology to a larger organization. And finally, government regulations. Across the world, regulatory bodies are working together toward harmonization so that if you have a product in Thailand, that you can also meet the regulatory requirements in Singapore, for example. And these regulatory bodies are also working to fast-track approvals so that it doesn't take as long to get a discovery or a therapy to market for approval. So with all of that excitement, let's turn to the meat of the talk. What is GMP and why do we have GMP? Before I go to that, I have one last poll question. Are you, listening to this talk today, are you working in a clean room? And so your choices are A, no, but you plan to in the next 2 years. B, yes, you currently work in a clean room. Or C, you have no plans to work in a clean room. We're trying to learn about you as we share some of our information with you. It's a two-way street, so we want to learn about you as well. Okay, so in transitioning from to GMP, it's a little bit different than academic research. So in academic research, the goal is knowledge, discovery, learning about a biological function or disease. Along the way to that discovery, you may develop a patentable invention or intellectual property that you then decide to commercialize. And that compares to GMP, where the goal is a treatment or a drug or perhaps a method or process that will be used in a human patient. So the goal is very different in GMP, in industry. It's not just knowledge itself to better understand a biological disease, but instead you're working on a product or a process to be used in a human patient. And that in and of itself is going to require greater safeguards. So that's why we have GMP. All right, so with all of this, what is GMP anyway? Let's define that. So GMP stands for Good Manufacturing Practices. And good manufacturing practices have a goal of making a product or a process that is safe and pure, that is effective, and that is of high quality. So GMP, good manufacturing practices, are simply minimum requirements that a manufacturer has to meet for this product or process. These are only guidelines. They are not step-by-step rules. And so there are many ways to comply, and it's different for every process, every institution. Now, typically, you would ensure your quality of your product through an effective quality management system, and I'm not going to get into that too much today. So when thinking about GMP, a major goal is to prevent contamination of that product or process. So in exploring why we need GMP, I want to talk about cell culture contamination. So one type of microorganism, or one type of contamination, and the biggest type we think about, I think, are microorganisms, including viruses, which may be latent or oncogenic in cultured cells, bacteria, including mycoplasmas, which are the most common cell culture contaminant, and fungi, yeasts, and molds, which are can be a problem in clean rooms. Another type of cell culture contamination are competing cells. This is when you've got cross-contamination of a related cell type or similar cells that are introduced by accident and contaminate your final cell product. And then finally, volatile organic compounds. These can be laboratory cleaners and disinfectants, which are sometimes used too much to excess in production. And so there's definitely an important consideration for use of laboratory cleaners and disinfectants, but it's also important to be aware of how those compounds can cause toxicity and cause contamination of the cultured cells for an end product. So where do these microbial contaminants come from? Number one, most importantly, they come from us. They are normal flora on our hands, our breath, our hair. Swabs of human skin, a study was done in 2008 by Elizabeth Grice and her colleagues. They found that 10,000 microorganisms per square centimeter is present on all of us, on our skin. The other source of microbial contamination is from the air around us. This is why many of us are working in clean rooms and why I asked that question. Even normal indoor room air contains between 30 and 1,000 microorganisms per cubic meter of air. So you can see that's a huge risk. So how does contamination happen? And I'm going to take you back down to a little bit of my academic history. How does contamination occur? It occurs when your aseptic technique deteriorates. So here I'm going to take a few minutes and talk about kind of a story in my life. So the x-axis here is measuring the time over the course of 1 year, 52 weeks, of what I'm measuring. And I think of this in terms of I've just started a new project and it's really exciting. We've got it all planned out. We're going to get great, clear results. I'm going to get published in Nature. And so I'm very excited. And at the start of my project, time 0, I'm measuring the quality of my aseptic technique. And you can see I'm being really careful. The quality of my technique is nearly 100%. Now, I think I mentioned this is in an academic environment. So at the start of my project, I also have a new undergraduate student working in my laboratory. His name is Mike. And it's his job to clean the laboratory and the lab equipment. And it's Mike's first lab job, and so he's really excited, and he's doing a great job. As you can see, the bottom of the graph here is measuring the amount of circulating contaminants in the lab, and it's very low. And now a few weeks in, my friend Stephanie comes in and starts talking to me while I'm working with my cells in the BSc, and she starts telling me about this good-looking guy who's joined the lab down the hall, and I start laughing, and I touch my pipette tip to the outside of my growth media bottle. But I catch myself. I make that mistake, but I discard that pipette tip, and again, my technique goes back to really good, nearly 100%. Now, it's midterm of the semester, and Mike skips a little bit of time coming to the lab to work because he's studying for his midterms. And so we have a transient or a temporary buildup of dust and dirt in the laboratory. And now time goes on. We're several weeks into my project and things are not going well. I'm having to repeat experiments. I'm not getting the clear results that I expected. My principal investigator has put another project on top of me, and I'm really feeling crunched for time. I'm starting to skip steps. And at the same time, Mike is figuring out that cleaning laboratory equipment is not quite as exciting as he had thought, and so he's not doing as good of a job either. And so this trend continues. And eventually, there's so much dust and dirt in the lab, and I'm skipping so many steps that these two lines cross, and that's how you get a contamination. And so what happens? I have a little talk with myself and remind myself that I cannot skip those steps. I have to wipe the BSc work area down with disinfectant before I start working with my cells. And I have a little talk with Mike and tell him, Mike, it's really critical that you do a really good job of cleaning the laboratory because everything we do depends on your work. And so the cycle begins again. So why do we implement GMP? This is why, because the goal of GMP is to make a product that is safe and pure, that is effective, and that is of high quality. And so the goal of GMP is really to reduce that human variation in a system. So the parts where I'm skipping steps and Mike is not doing a very good job, GMP is designed to eliminate those problems. GMP is designed to ensure that each task happens exactly the same way each time, regardless of who is doing that task. And ultimately, GMP is there to protect patients and to ensure that the product is safe, is pure, is effective, and is of high quality. So what is GMP and why do we implement it? Number one, it involves defining the practices and protocols with clearly established procedures that everybody can understand so that those practices are repeated exactly the same way each time. If there are any changes, we clearly explain what those changes are and why we made that change. And then finally, detailed training for the people working in this process, and like extensive training so that everybody is performing a task exactly the same way every time. And that training is renewed periodically as refreshed so that people continue to have that high level of practice. GMP also includes documentation, documenting all of those steps as they happen. So it's not like in an academic lab where you might make a note on a paper towel and then attach that paper towel in your laboratory notebook. No, that does not happen under GMP. So the exact procedure is followed, that is signed with a name and a date, lot number, batch number, and what the results were. And then finally, GMP is about record keeping. The goal of the records is that anyone can understand what was performed, why something was done, and that is clear and trackable, and that record is easily searchable. So that would include an index, for example. So you can run down the index and find a given task on a different day and how that was performed. So this brings us to the key GMP principles as compiled by the International Society of Pharmaceutical Engineers. First of all, as I've said, GMP is about step-by-step procedures and work instructions to really prevent contamination and mix-ups. That's the goal, to keep everything at a high level, repeatable, and to prevent contamination, anything that could endanger human health when your product is released to the market. GMP number 3, it's about promptly and accurately writing down, documenting what happened so that you can comply, so that there is traceability of what happened in producing that therapy or drug. Number 4, GMP is about validating the system. Everything is doing what it was designed to do. And number 5, that extends to the facilities and the equipment. And number 6, exactly how to maintain the facility and that equipment. Number 7, job competence, training. That's really critical, that everybody is trained exactly so that everybody can work exactly the same way. And number 8, again, to protect against any contamination because everything happens the way it should. Number 9, it's about building quality into the product, controlling how models are used, validation of the process. How does the sourcing happen? Is the manufacturing controlled, even down to labels and testing? And then number 10, the big one, conducting periodic audits to make sure that everything is happening the way it's supposed to. So all of these things go into the manufacturer of a product implementing these procedures so that they can pass audits so that they reach compliance. Now, it's up to the user to create their own GMP system. I can't tell you what procedures and records you should do in your institution. GMP principles are only guidelines because each institution is different. You have your own personnel and processes. Your training is going to be different because of that. You may not have the same equipment as a company down the road. Your facilities are certainly going to be different. Your documentation is going to be different because you may start with, you may need slightly different information than a different institute might need. And finally, your product is different. The good news is you know your systems like no one else can, and you can build on that knowledge within the GMP framework. Now, there is good news. Laboratory equipment can help you on your road to compliance. So let's talk about that. So laboratory equipment is the center of your production for regenerative medicine. And so what that means is that your equipment must be your GMP partner. So your equipment has to meet given performance needs so that your cellular product meets the viability and quality requirements that you set out, so that it's reliable and repeatable, so that you're minimizing lot-to-lot variation, you're minimizing that variability that is inherent to biological systems. Minimizing that variability is really the goal of your laboratory equipment. If you're talking about an incubator or a freezer, you want fast recovery from a door opening so that your cells or your samples, your frozen samples, are spending their maximum time at their ideal conditions, and that there's uniformity throughout that chamber so that if you put your cell cultures in the top of the incubator, they're going to be experiencing the same conditions as if you put them in the bottom corner of the shelf. Number 2, your lab equipment must offer clear documentation. This means more than just a user manual. I can't tell you how many people I talk to working under GMP or in clean rooms who haven't read the user manual. And that's a shame, because you can learn a lot from reading the user manual. Also, it's not just about specifications. A lot of freezers might have the same type of temperature probe, but it's not about the specifications and accuracy of that temperature probe. It's about how does that probe behave within the framework and the design of that freezer. What's the air circulation like? How big is the chamber? Things like that all figure into the performance. Maintenance, how do you maintain that equipment? Does the user manual tell you? And finally, control. Can you lock out the controls? And then the last section, that equipment should be easy to use. Everyone should understand how to operate that equipment, how to work the menus. It should be easy to train on so that you can describe it and people get it right away. You don't have to keep repeating that training just to get people up to speed. The equipment should be easy to clean. This is critical to limit contamination in the work area. There should be clear monitoring and data logging, and we'll talk about that in just a few minutes. Now, the other good news is all of these needs may be met by the laboratory equipment you already use. So let's talk about what you can expect. What are some important considerations for GMP lab equipment? Number one, cleaning and disinfection. This is central to GMP. Make sure you find lab equipment that provides you clear recommended procedures for cleaning and disinfection. The manufacturer should be able to offer recommended disinfectants that are compatible with the materials used in that laboratory equipment so that you know clearly which disinfectants are okay, that are not going to corrode the stainless steel, for example, that will not, work with the metals in your equipment to cause some sort of volatile organic compounds or toxicity to your cells. You should expect a clear and complete list of the materials used so that you understand clearly how the disinfectants that you use might react. All of those things are designed to aid your cleaning, designed to control contamination in your processes. They should be compatible with different types of vaporized hydrogen peroxide, and finally, glove-friendly controls. And the US FDA just in the last week or two came out with new guidances in response to the pandemic, requiring use of gloves and glove-friendly controls, and requiring adding additional requirements for cleaning and disinfection. So these topics are really important right now. I mentioned materials composition. Here's just an example of a list that you could expect to receive. I mentioned compatibility with disinfectants. So here's a list, for example, that you could expect of disinfectants that are okay for use with given equipment, and also, importantly, cleaning compounds and disinfectants to avoid because they will cause corrosion or damage your laboratory equipment. Maintenance and qualification. This is huge when you're using laboratory equipment in a controlled process. What kind of checklist is there for installing this equipment? Can you get guidances on installation qualification, operation qualification, performance qualification, temperature mapping? Are those documents, is that guidance available to you from the manufacturer? Are you able to confirm the calibration of your equipment, or can you correct that calibration, and are there clear methods for doing that? Can you test the alarms and alerts? Do you understand why you have those alarms and alerts? Again, training, can the manufacturer help you with that? Maintenance checklist, do they provide you with routine maintenance? What's expected every 6 months or every year? What spare parts should you have on hand? And finally, if you need help, can you get it by customer support and technical support? I mentioned performance being more than a list of specifications. You should look for recovery from a door opening. How fast does the temperature recover from a given length of time of door opening in an incubator or a freezer? You should look for uniformity, top to bottom, side to side. They should be able to provide you with a map that shows how that design should behave in a given scenario. Factory acceptance test documentation. What happens at the end of line testing for that equipment? Are there certificates of conformance that you can see and that will get you along your road to compliance? Are there design performance examples? What tests were performed? What were the results of those tests on your given serial number on your equipment? is there end-of-line testing that has been performed on the units that you purchase? And can you verify what happened in that end-of-line testing? And then finally, of the features offered on that equipment, a sterilization cycle, for example, or biocontainment lids on a centrifuge, were those tested independently by a third-party laboratory? And can you see the results of that testing? All of this information may not be available from all manufacturers, so this is why I'm suggesting that you ask these questions, because you should have this information. It gets you a long ways along your road to compliance. And then finally, I mentioned documentation. So what features are validated for performance? Are there regulatory certificates? If so, you should have copies of those. Quality management systems certificates. Make sure you can follow the user manual, and so on. Specifications, drawings of the equipment so you know how it will fit into your clean room facility. Can you get it through the door? What's the footprint like? Data logging and downloading. And now I'm going to hand it over to my colleague, James.
Hi, morning to everyone. Thank you, Mary Kay, for such an insightful presentations. I'll take over on the part on data logging itself. So what's data logging? Data logging is a process of collecting, storing data over a period of time in order to analyze specific trends, record database events, actions of the systems, network, or IT environments. It enables the tracking of all interactions through which data, files, or applications that are stored, accessed, modified on the storage devices on applications. With current demand for data and gaining recognitions on how data can be studied, analyzed to understand and improve processes for both individuals as well as companies, there is an increasing focus on how data can be collected better and better utilized. Here is an example. Here is an example of a temperature log card that's used by CDC US to monitor the temperature of the refrigerator. Typical temperature of storage devices are checked and recorded either manually, at least twice a day or more, or by using an automated systems via wired or wireless monitoring systems in their facilities. Currently, in the current COVID climate and manpower crunch, we are seeing a shift towards automatic systems such as wireless monitoring across various laboratories to ensure productivity and efficiency. Here are some examples of available data loggers in the current market. In Singapore, across various labs, I would find data loggers ranging from basic chart recorders to standalone digital data loggers that you've seen on my left. These data loggers are widely used to monitor lab environmental conditions such as temperature, humidity. Data are collected manually on a fixed routine by operators. The data will then be uploaded via data entries or stored by filing itself. This is considered a highly labor-intensive process. Going forward, as was mentioned earlier, with wired monitoring systems, Mary Kay, sorry.
I'm sorry, James.
Okay.
There we go.
Yeah, so with increased focus on automated systems, we are seeing wired monitoring systems such as building management systems, BMS, that are generally implemented in bigger corporations for data collections in order to adhere to compliance standards and their required processes. Again, environmental conditions and additional parameters such as CO2 and diffusion pressures are added so that they are able to ensure the right parameters, to ensure the right airflows into the clean rooms can be monitored. Given that there are wider varieties of sensors and easier deployment with automatic systems, we are seeing increasing focus towards this. In recent times, an improvement in technology we are seeing again a shift from wired to wireless because this is easier, and again to increase our width and lesser cost, lesser investment, so that they are able to enhance the productivity of data collection as well as management. Next. So why do we data log? Here are some examples. Data logging supports samples protection. There is never a 100% guarantee that all equipments are always good in good working conditions. Numerous factors can cause system failures will occur. Having a reliable data logging system will be able to enhance your sample protections with instant alerts for our range sample conditions. These alerts will also enable urgent proactive response from the team itself. Secondly, data logging support asset and cost optimizations. A remote monitoring system will serve as part of your assets management strategy by minimizing costs for insurance policies that covers replacements of equipment and products. With effective data collections and effective data management, predictive maintenance strategy of your equipment can be implemented. This will lead to reducing costs of maintenance, less downtime of your processes, and of course, higher productivity. Lastly, data logging support compliance. Automatic and continuous data logging creates a permanent audit trail to support compliance standards and consistent, reproducible results. In the next few slides, we will share a variety of various regulatory requirements that focus on the need for a good data logging systems. As discussed throughout this process, a reliable and effective data logging setup is very much in line with the GMP guidelines. So basically, as mentioned, we will be sharing some of the guidelines. This slide contains extract from WHO, GMP, and GLP guidelines. Electrical supply, lighting, temperature, humidity, and ventilation should be appropriate and such that they do not adversely affect directly or indirectly either the pharmaceutical product during the manufacture or the storage or the accurate functioning of the equipment. Basically, what it means that the parameters itself that governs the environment, the parameters itself that governs the storage, as well as the production itself should be monitored. Next. Some further extracts from WHO guidelines itself. Here, we zoom in on the environmental factors such as temperature and humidity, measurement frequency, sensors accuracy, calibrations, positioning of sensors, as well as a set of data record management are clearly defined as recommendations for those who are keen to set up a GMP or rather a WHO GMP compliance lab. What we are seeing here are various parameters defined for temperature as well as humidity, basically setting you up for the right guidelines to work when you want to do work in the lab itself. Next. In this slide, we further list the GMP guidelines for storage, such as temperature controlled rooms, cold rooms, refrigerators, freezers requirements. Besides temperature and humidity monitoring, power stability and alarm requirements are also key to a good GMP storage. Many often we just basically observe, many labs basically monitor the basic things like temperature and humidity. With more and more focus, we are realizing that as you have more and more instruments installed in your lab, power to individual instruments are vital, especially those storing vital products itself. Hence, power stability, alarm requirements are now put in as one of the key requirements or as one of the key guidelines under the WHO guidelines itself. WHO guidelines also indicates the parameters for transportation of time-sensitive products. Next. Another indications of importance of effective and efficient data logging. From the extracts that we drawn out from College of American Pathology, CAP, it highlights the importance of a proper calibrated storage equipment as well as the type of temperature monitoring to be used. Again, alerts are indicated to be established for each measuring devices so to allow adequate and accountable time for response in case of any emergency. With this in place, I would say that operators or the lab users itself will be able to react in time to prevent any major downtime of the equipment or to prevent any major disruptions to the product or major impact to the product itself. Next. Another key regulatory guidelines is under USP, US Pharmacopoeia. Under this regulatory guidelines, it defines the control environment. Things like temperature mapping, proper calibrated temperature monitoring equipment, as well as the need to have the mean kinetic temperature parameters instilled in the process. MKT, which is mean kinetic temperature, is a simplified way of expressing the overall effect of a temperature fluctuations instead of just monitoring the regular temperature value. From experience, MKT is a simplified way to express the overall effect of temperature fluctuations during storage or transitions of perishable goods. MKT is widely used in pharmaceutical industries. From experience again, MKT itself actually should be used in situations where control is good and where occasional excursions may be encountered. So what it means basically, if you have a set of very good systems that set up with the proper guidelines, with the proper compliance, MKT itself provide you rare occasions where it happens. However, if you do have and if you have poor temperature control for storage or transportation devices, MKT itself might not be that effective. While it's not possible to obtain a meaningful MKT value from daily readings or of simple max mean temperature thermometers, such as temperature fluctuation, it is noted that some data loggers and building systems are capable of recording multiple temperature readings over a period of time and offer the functions of calculating MKT itself. So again, this is important. It's a parameter that is encouraged to have when you have proper temperature controls. Next. Finally, with huge inflows of data, you need a strong data management and protections. This is where US FDA CFR Part 11 comes in. It is a key regulatory requirement to govern data management. A good monitoring system should have the software components that creates, modify, maintain, archive, collects, and transmit electronic record that may be required by FDA regulations. The system should have electronic signature, but not biometric electronic signature of any kind. Person responsible for controlling access to the content of the electronic records may not access the system's internal database, internal module memory itself, or basically containing said records. Main concern is the system itself should be a closed system. As stipulated in 21 CFR Part 11, the definition of a closed system is as follows. Closed systems defines that environment in which the system access is controlled by persons who are responsible for the content of electronic records that is on the system. Please note, while it is possible to download certain parameters for sensors during setup, the users itself must provide authentications each time they log in to perform any task. In addition, system owners may take appropriate measures to protect access to record content, as well as to protect the record content itself so that it may not be modified, destroyed, or otherwise compromised by unauthorized users, improperly trained users. Basically, every single action that you have to undertake for your systems itself has to be recorded and traced. Here are some examples that I would like to highlight when you have, I would say, deviations to controls. Here, I basically indicates temperature. Temperature is generally considered the single most important factor for determining product quality and safety. This definition means that a lot of other process parameters or storage conditions may more or less influence upon products in different situations. But temperature is actually the main physical value, and the impact is always erroneous. Here we have two published articles that highlights the impact of temperature. We've seen impact of temperature to vaccines. We've seen temperature impacts to blood. In this article, we've seen that it's clearly stated that temperature fluctuations impacted the quality of the product, render it ineffective, and then has to be discarded. It applies for both vaccines and blood itself. In this situation, due cost and of course impact to the situations, to the productions, or to the people who require this are incurred. Hence, we will never be wrong to say that proper temperature control, and again, temperature control is the prime single receipt for success. The temperature control whole supply chains for refrigerated processing, storage, distributions, retail, and household handling of products such as cell is therefore paramount importance for guaranteeing safety, quality, wholesomeness, and extended shelf life of perishable commodities. Next. As mentioned earlier, one of the most effective, effective ways of using data itself is to be able to have an effective and predictive maintenance cycles. Here we have examples where the users is able to identify problems using the data they have collected, they analyze it, and they determine that there is a potential problem that comes about. Maintenance can be planned in accordance to this data, and then we can have earlier shutdown, we can have a proper plan in place during the shutdown, and because of this, it's been executed more effectively, and unnecessary downtime are being avoided. As such, with data, it allows us to have better planning, better executions, more organized management, and of course, in the end, lower costs and higher productivity. Next. To sum up, with effective data collection or effective monitoring, basically you'll be looking at enhanced protection of the quality of the samples, better reactions, maintenance schedules with your various data. It allows you to have various alarm notifications, and of course, lastly, compliance to regulations, compliance to GMP guidelines itself. Again, this allows us to fit compliance to the GMP guidelines, will give us better organized management and of course higher productivity. Next. So to sum up, here's basically our Thermo Fisher Scientific sum of our product offerings itself. We do have a series of GMP focused products with the necessary documentation package as well as the required compliance service. Here we are able to offer you the full suite of solutions from equipment to documentation as well as to the services. Thanks. And finally, we are committed, to our scientific enhancements. with products and solutions that enable customers to push the boundary for innovations. The key things that we'll definitely be looking at always with customers focus. We have unmatched portfolios of products. We are leading industries. And of course, we have a depth of capabilities throughout our organizations that help you, fulfill the requirements for the GMP guidelines. With this, I end today's presentations. Hope you have enjoyed our webinar sessions. Over to you, Lydia.
Thank you, Mary Kay and James, for the presentation. So we're now ready to take your questions. If you have a question for our speakers, please send it through the Q&A box, which is indicated by a question mark icon near the bottom of the screen. All right, Mary Kay and James, we have got 3 questions that has come up. So I'm just going to read this through. Can you talk about your recommendations for cleaning lab equipment, especially for the coronavirus?
I'll take that one, James. Okay, so cleaning lab equipment, especially for the coronavirus. This is a question we've been getting a lot. First of all, start with mild dish soap and water. Many studies have shown that the coronavirus is very sensitive to soap and water. And then after that, follow with a disinfectant such as 3% hydrogen peroxide, no more than 6% hydrogen peroxide. A quaternary ammonium disinfectant is good for all of our stainless steel and glass in our laboratory products. You must avoid chlorine-containing disinfectants such as bleach. That will cause corrosion to the stainless steel and will cause bigger problems for you down the line. Bleach is not needed to kill the coronavirus.
Thank you, Mary Kay. Second question that came in. I'm working with iPS cells for cell therapy. What would you recommend for specialized lab equipment?
So that goes back to performance. So it's not just comparing the list of which incubator has which sensor, but really, how does that incubator perform? How fast does it recover from a door opening to your set conditions for 37 degrees and 5% or 8% CO2 and high humidity to prevent evaporation from your incubator? I would also look for proven features such as a sterilization cycle on the incubator. I would look for biocontainment properties for a centrifuge. Depending on your scale, I would look for features that ensure reliability and repeatability so that you minimize lot-to-lot variation. And thirdly, monitoring for your cells is going to be critical. So that takes us back to what James was talking about for data logging. James, do you want to add anything there?
Basically, you covered everything itself. Again, as I said, data logging, collection of data itself should be implemented so that you have the necessary data itself to do the analysis after you do the setup or after you finish the processes itself.
Right, and I would add that we're happy to help you answer more specific questions about laboratory equipment. We've got a wide array of Thermo Fisher Scientific people here waiting to help you.
All right, thank you, Mary Kay and James. So next question that came in from Lee Yong Chi. For lab equipment, it seems like only IQ, OQ, PQ is needed. Is FAT necessary?
Oh, FAT as in factory acceptance test. I think I would call that an indicator of the quality of your equipment. So what kind of testing at the end of the production does your equipment undergo? Are all of the parameters tested? Are the electronics confirmed to work the way the manufacturer expects them to work? Are the features tested? So, for example, at Thermo Fisher Scientific, 100% of our CO2 incubators and ULT freezers undergo end-of-line testing. So all of the parameters are tested. If it has a sterilization cycle, that is run. And then there's a final repeat review of the packing list before anything is shipped, for example. Those are just a few of the examples of the tests we run.
Thank you, Mary Kay. I think the question is for SAT, as in site acceptance test.
Sorry.
Okay, that's fine. I think it's good to know about the SAT as well. Good.
Is that one for you, James?
I'm fine. I'm fine.
Okay. I guess I'm not familiar with that term. I apologize.
Sure. All right. And next question. Can you recommend someone to help me with GMP?
Well, I would probably start with our CTS series products, which come with all of that documentation that we were talking about this morning, and then also contact our compliance services people who are experts in that, and we can share some contact information there following the seminar.
Thank you, Mary Kay. So I think we have addressed all the questions that has been asked. If you have a question for our speaker, please feel free to type it in, and we can still address it after the webinar. All right, so I think we're right on time. Our next talk will start at 10:15. So we're going to end with a first. Over to you, Sonia.
Hello, good morning, everyone. Hi, Lydia. Can you hear me?
Yes, I can.
All right, great. So thank you for joining us this morning at Gibco Galaxy Tour Part 2. So right now we'll proceed for a short poll. So on the right side of your screen, you will start to see a poll popping out. And for this poll, there will only be 3 questions. Namely, do you think having an audit trail is important for a GLP compliance lab setup? Second, do you think temperature monitoring is critical for your GLP compliance processes? And the last question, are your processes currently FDA CFR Part 11 compliant? Okay, so after you guys have completed these 3 questions, you guys can go for a very short bio break or coffee break, and then we'll come back at 10:15 a.m. And please participate in this poll because you will also stand a chance to win our lucky draw prizes, which we'll be announcing on 14 October. We are going to give out the Logitech headset, which you guys can use for your scientific presentations. And also we are giving out a very exciting UV sanitizing box plus wireless charging feature. So don't forget to participate in all the polls. This is going to be our 3rd poll, and there will be another poll later as well. So if you participate in all the polls, you'll stand a higher chance of winning the lucky draw prizes. All right. And if you have any other questions that might come to your mind after the presentation from Mary Kay and James, feel free to type into the Q&A box. Our panelists will still be here online, so don't miss the opportunity to ask questions. All right, there's 1 minute left for the poll. So for those who have not completed, we still have some time. For those who have completed the poll, thank you very much. You guys can go for a short break before we come back to Jacqueline's talk about to launch our Gibco rotier system. All right, everyone, the poll will be closing in 10 seconds time. So if you're at your last question, please complete it. These are multiple choice questions, all right? Okay, thank you everyone for joining the poll. We have ended the poll. So see you guys at 10:15 a.m. Stay with us and we'll be back at 10:15 a.m. Thank you. Good morning. Thank you for staying with us. We'll be starting in 5 minutes time. Good morning, everyone. Once again, welcome to Gibco Galaxy Tour Part 2. So we'll be starting in 2 minutes time. Thank you for joining us so early this morning from Singapore 9 a.m. to Southeast Asia 8 a.m. in the morning. The next few topics will be very exciting, so please stay on with us. And very soon we'll commence. Thank you.
Hi everyone, welcome back. I hope you enjoy your break. We will now be moving on to our second presenter, Jacqueline Ng, who is our Regional Manager for Cell and Gene Therapy. In this session, Jack will introduce our new Gibco Rotia CTS counterflow centrifugation system, which is designed to enable scalable and cost-effective manufacturing of cell and gene therapies, and to allow for the seamless transition from research to clinical and commercial cGMP manufacturing on the same instrument. Jacqueline will first present this in English, and at 11:30, which is about 1 hour and 15 minutes from now, our product manager, Jasmine Sun, will be repeating the same presentation in Mandarin. For the benefit of those who just joined us, please feel free to send your questions through the Q&A box at the bottom of the screen. And now, over to you, Jacqueline.
Hi everyone. So can you hear me well, Lydia? I'm just having a problem with turning on my video, so I don't think anyone can see me.
I can hear you well, but yes, I cannot see you on your video.
Okay, just let me fiddle around with this a bit, and maybe I'll just take away the virtual background. Okay.
Yeah, can see you now.
Okay, might be the background. Okay, so I'll start. And so thanks everyone for joining us today. And I'm Jacqueline, and I look after the cell and gene therapy business in our Asia Pacific region. So for those of you who were here earlier, Mary Kay and James gave a really nice talk and some context on what it is to what is GMP, when and why it's needed, and what it takes to implement GMP processes. So in the next two talks, Jasmine and I will shift gears a little, and we're going to talk about moving towards using closed systems in cell and gene therapy processing. And I'm very excited, to be given this opportunity to introduce the Rotia. The Rotia is an innovative system, and I believe that it can bridge some of the current gaps in the field today. And I'll also touch on some of the industry challenges and other technologies that we have that will be essential for the field. Now, if you have any questions, feel free to write them in the chat box, and we will have about 10 minutes for Q&A after my presentation. The cell and gene therapy field is rapidly evolving, and we have seen recent successes in clinical outcomes, increasing landmark marketing approvals, and investments by both public and private sectors. However, the technologies and processes around cell therapy manufacturing are still catching up with the market demand. An analogy I always like to use is that cell therapy manufacturing today, it is like flying a plane while building it at the same time. The engineers have the skill but lack the critical parts to build the plane. And in some cases, parts do not exist at all. Now, listed in this slide here are common challenges that we see in the industry. Cell therapy developers have to ensure that therapeutic products are consistent in quality across the entire manufacturing workflow, and also consistent in quality across batches as they scale up and scale out to other countries. In yesterday's talk, my colleague Ryan spoke about some considerations in how one would perform identity, potency, and safety testing. So if you did not have the opportunity to attend that talk, we could follow up with some slides and recording of that webinar. Another challenge is meeting regulatory compliance. This is not an easy task at all. Ensuring that every single raw material that's used in manufacturing comes with the appropriate traceability, documentation, and QC testing. It's really a very daunting task. And in some cases, GMP-grade reagents do not exist at all. Ideally, when you want to manufacture cells for clinical trials or commercial manufacturing, we should use GMP-grade reagents, but in most cases, they don't exist at all. And most cell therapy developers typically come to suppliers like Thermo Fisher to customize RUO reagents such that it meets local regulatory requirements. And any customization, addition of QC testing can be very costly. That is why we always encourage the field to source and validate processes of GMP-grade reagents as early as possible, during the preclinical and early phase trials. So this would eventually accelerate speed to market and overcome regulatory barriers down the road. And now one of the biggest challenge that I'm going to touch on today is how to help the industry bring down cost of production, specifically cost around bioprocessing. And a way to do so is to move towards adopting closed modular systems, which can be automated and controlled remotely through digital integration. And now let's take a deeper dive on some of the main issues in cell therapy manufacturing. And we are giving an example here with manufacturing an autologous T-cell therapy. So if you look at the workflow in red, the red boxes, after leukapheresis, cells will undergo isolation. It could be T-cell isolation or PBMC isolation. They will undergo activation. This could be done through viral or non-viral methods. Expansion, harvest of cells, a media exchange into a cryopreservation media or formulation media such as injectable saline for administration. Again, this is a complex process, and today it's very common that a lot of users use open processing, that involves centrifugation, opening and closing tubes, doing culture with flasks in a biosafety cabinet. So the layer of complexity comes when we try to synchronize and harmonize different instruments, different reagents and processes, and to make the entire workflow trackable. It is very challenging to document every single step if the processes are manual today. And based on these issues, we understand that cell therapy developers are looking for three main solutions. The first, to close the system and close the processes and minimize open manipulations that could potentially introduce risks of errors or such as contamination. Cell therapy developers are looking at modular systems, separate modular units that are closed, so that they have the flexibility to use different units for different clinical pipelines or different parts of their process. And finally, cell therapy developers want to move towards automation to reduce labor and human errors. And now looking at the cost perspective, if the field were to apply open and manual manufacturing in a GMP facility, investment in Class A and B clean rooms would be required, and this demands a lot of investment in infrastructure, environmental monitoring, and labor. And that is why the field is moving towards cell processing in Class C clean rooms, and this entails using closed automated processes, and eventually this will achieve significant cost savings. So the largest reduction is cost savings. It's really around labor, utilities, and the infrastructure set up in a GMP facility. Now, given this backdrop, what our company is doing is building out different closed systems combined with GMP reagents that exist today and optimizing protocols together with our downstream analytical solutions, putting them together and making a fit-for-purpose cell and gene therapy manufacturing platform for different types of therapies. Well, we can physically integrate all these systems. We are also enabling digital integration. And as an example, our goal is to provide direct connectivity to Emerson's Delta V platform, which is 21 CFR compliant, and this would enable full automation and allow features such as batch record keeping along the entire process. And the brand that most of you would might be familiar with is our CTS brand. That's our Cell Therapy Systems brand. And I'm introducing this CTS brand, because I want you to understand what comes along with this package if you were to use any of these reagents or instruments that has the CTS brand. Now, the Gibco brand of products is thermofisher's GMP grade raw materials that are specifically designed for cell and gene therapy manufacturing. These reagents are well characterized. They're produced under and manufactured under our GMP facilities. In the US, we have US FDA-registered sites that are ISO 13485 certified and 21 CFR Part 820 compliant, and we follow USP guidelines as well. But more important, the CTS branded products comes along with very comprehensive documentation package, which includes various QC testings that has been performed, traceability studies, and in some cases, most cases, DMF filings. And in countries that do not support DMF, in Asia Pacific, our regulatory team provides regulatory support filing. And this would allow you to facilitate, IND filing when you're ready to proceed with that. And finally, the CTS branded products have proven use in over 200 clinical trials, and some of our products are already used in commercialized cell and gene therapies. So now, I want to introduce one of our recent innovation, the Gibco CTS Rotator counterflow centrifugation system. This benchtop instrument is the world's first benchtop counterflow centrifugation system, and its main purpose is to help you to wash cells, concentrate them, do media exchanges. Counterflow can enable cell size separation, all within a closed process. So these are some of the key top 4 features of the Rotator. And the first main advantage of Rotator is that it allows process flexibility. And I'll show you some pictures on how our software and protocol builder program looks like. The end users are empowered with the software tool, and you can create any types of protocols that you want without having to pay additional software licenses. So you could use the Rotator to create protocols for T-cell, washing T-cells, MSCs, or different cell types for many different applications. And we are also not restricting the types of bags that you use on the Rotator. You could use different bags, for different purposes and different types of media. The second key feature of Rotator is that it enables very high cell recovery and viability. For basic wash concentrate protocols in the system, we see over 95% cell recovery, which is very high. And the third key feature of Rotator, which I would say is the most important feature, is that it allows concentration of cells down to very low output volumes, as low as 5 mils, in very high cell densities. So we could harvest cells as low as 5 mils in densities as high as 300 million cells per mil. Some of our early adopters have even pushed this to 350 million cells per mil. And there's currently no commercially available closed systems that could achieve this low output volume. And this is very suitable for autologous therapies. We often don't get a lot of samples to work with. And finally, the last key feature of Rotator is that it's suitable for research process developers or researchers in preclinical phase could use it. It's affordable, and this same unit can also be used, in the commercial manufacturing. The single-use kits on the Rotator, it's closed, and the software features comes with an OPC-UA interface, which enables connectivity to 21 CFR Part 11 compliant systems. And this is how the complete Rotator system looks like. It comes with the instrument. In terms of the footprint, it's not very large. It's only about 20 kilos. It's very compact. It comes with a single-use kit, which is universal. So you could use the same kit for different types of processes and across different cell types. And to connect the kits, you could use tube welding. It's the industry standard in the field when working with closed systems. You use tube welder, or in a process development setting, you could attach lure fittings to the tubes to connect different types of bags. And it comes with a touchscreen tablet, which features the software, and you can visualize the entire process in real time with our high-resolution camera. So in terms of what the Rotator does and where it can fit into the cell therapy manufacturing workflow, in the typical cell processing workflow. You would collect your cells, isolate, activate them. You may or may not engineer them. You would expand them, and then you would cryopreserve them. So the rotier could fit into all these different steps. You could use the rotier to wash cells that are previously thawed. So our R&D has a lot of experience with washing frozen thawed leukopak samples, and we've done a lot of work with frozen thawed PBMCs or even MSCs. And for working with T cells, if you want to output in low volume, you could use the rotier to do a media exchange in electroporation buffer for a non-viral transfection of your gene of interest. And then you more commonly we see rotier used for washing expanded cultures. A common question that I often get is that what's the maximum volume of cultures you can fit on the rotier? So we would often say there's no maximum volume. It's a continuous batch continuous processing. So as soon as the counterflow chamber is full of cells, we would just harvest the cells and bring in your samples continuously. So we've done some processes with 5 liters of cultures down to 1 liter of cultures. And we have early adopters from biopharma companies who are working with 10 liter batch sizes on the single instrument. And I won't attempt to play the videos on this WebEx platform, but I believe my colleagues would have posted 5 links to YouTube videos on rotier, and you can open it and view them at the same time. Okay, so before I go into applications, many of you might not be familiar with what counterflow centrifugation is about. Counterflow centrifugation is not the same as typical centrifugation. When you do centrifugation in a normal centrifuge, there's only one force that is applied, which is G-force, and then you get a cell pellet. So when working with counterflow, you have to shift away from the mindset of cell pellets. So with counterflow, you would have a counterflow chamber, and on the rotier kit, this chamber is the 10 mil chamber. The size is not very big, but we could fit in about 5 billion lymphocytes in that small chamber. And with counterflow, this is what happens. So I'll go through step by step on how we would process cells in the rotier. So first, you would load your cells. So you have a bag of sample, and will come through the tubing. The process an opposing G-force, which will spin and keep the cells in a fluidized bed. So this is not a cell pellet, and we call this term a fluidized bed. So the G-force will keep the cells in the chamber, and the flow rate comes in at the opposing force. And this fluidized bed, it's similar to how you see cells in suspension. So in this state, you can do many things. So the cells enter the chamber, and they will exit through the base of the chamber. But to prevent the cells from leaving the chamber, we would balance the G-force such that the cells stay within the chamber. So at this state, you could do a media exchange. You could input a different media and wash the cells. You could do cell separation by size. And over here, we have cells that are the blue cells and the yellow cells. If we tweak the G-force, if we lower the G-force, what happens is that you can collect the cells that are smaller in size, through this process called eluteration, and cells will exit from the base of the chamber. And then you can keep the big cells behind, the blue cells, which could be, and this example could be, separating your lymphocytes from the monocytes.
Hi,
Again,
Jacqueline.
Mm-hmm.
Hi, Jacqueline. Sorry to interrupt. I think we're breaking off a little bit during the explanation. Would you mind going back to the previous slide again?
Okay, so where did I stop? Should I explain from the start?
Yes, from the start. You were breaking off quite a bit at the start. Sorry about that.
Okay, sorry, because I'm actually calling in from the office now, so I apologize if the network is not stable. Okay, can you hear me a bit better now?
Yes, I can.
Okay, so let me just start again and go a bit slowly. So in normal centrifugation, you spin at only one force, which is G-force, and you get cell pellet. So with counterflow centrifugation, the principle is totally different. So you need to put away the mindset of normal centrifugation and cell pellets. And with counterflow, you are dealing with two forces here. So we have G-force and flow rate. So upon sample loading, you would have a bag of cells sitting on rotator, and rotator would bring in the sample at a specific flow rate to an inner tubing of the counterflow chamber. The size of the counterflow chamber is about 10 mils. Okay, and at the same time, cells enter a specific flow rate. The chamber is spinning at a G-force in the opposite direction. So we have two opposing forces that keeps the cells inside the chamber. And when cells are in this state, we call them a fluidized bed. Alternatively, some would say that the cells are in suspension, but it does not form a pellet. So the G-force here is balanced with the flow rate. It keeps the cells in the chamber. The cells are there.
Hey, Jacqueline, I think we lost you.
Does not exit the chamber into. And this okay, so where did I stop?
about 15 seconds ago.
Okay, 15 seconds ago. Okay, I'm so sorry about that. Did I talk about the fluidized bed? Could you hear me explaining that?
About the fluidized bed, yes.
Okay, so when cells are in the fluidized bed, you can do media exchanges. And okay, so maybe I'll just stop the video. Right. So when cells are in the fluidized bed, you can do media exchanges. You can do a cell separation by size, and you can what we've done RBC lysis in the chamber as well. Okay. And again, this is not a cell pellet. Cells are in suspension. And over here, we have two color cells, blue cells and yellow cells, and we're just simulating the bigger cells from the smaller cells. So when we tweak the G-force, when we change the G-force or any one of these parameters, as an example, we are lowering the G-force. Okay. Okay, as we lower the G-force, we can allow some of the smaller cells to escape the chamber, and we can harvest the smaller cells and keep the larger cells behind. So this is a clear example of how we would do PBMC wash and separate lymphocytes, which are smaller in size, from the monocytes, which are larger in size. Okay. Am I clear now with my explanation, Lydia?
Yeah, I can hear you fine now.
Okay, great.
Thank you.
And just to repeat, this is what happens during cell separation. It's a process called, we should call it eluteration. So we can separate the smaller cells from the larger cells. In this case, we can separate lymphocytes from monocytes. And these are the key, again, a summary of the applications of what can be done in Rotair's counterflow chamber. So first, we can do cell loading in the chamber. And second, as cells are kept in the chamber, we could do media exchanges and washes. We can do eluteration. Another term that you would call this is separation of cells. And finally, you can concentrate the cells and harvest in very small output volumes as low as 5 mils. Okay. And now I would go through step by step on an example of how we would run and process a protocol. And again, my colleagues have included some links on the chat box, and you can view the links to this specific video after the presentation. So typically, we would set up a separation demo using beads. And on the rotator, you can see that there are 3 bags connected. On the right, there is a bag of green color beads, which is actually consists of blue and yellow beads. And on the left, this is a picture on how the protocol, the software looks like. We would have a picture of the kit, and the end user can take the picture and attach bags according to what this kit looks like. And in the first steps of every run, we would do a basic prime because the kit will be empty. So we need to fill the lines and the chamber with fluid. And the priming buffer that we use could be media, or it could be a wash buffer such as PBS and 1% HSA. And then, the next steps, we would continue the priming sequence, which takes probably about 10 to 20 seconds. We can dilute the sample. And now in this step, we are loading the sample at a specific flow rate. Over here, it states 40 mils per minute and a G-force of 2,700 Gs. So the maximum G-force that the rotator can run at is 3,000 times Gs. Okay. As the sample loads, this is an appropriate setting to load the beads. And this is how a stable fluidized bed looks like. You can see the beads in here, and we don't see any beads exiting through the base of the chamber. So this flow rate and G-force setting it's a good setting to keep the beads in the chamber. And at any point of time, you could adjust the parameters in real time. You could increase or decrease flow rate and G-force, and you could extend the steps as well. And then when it comes to eluteration, we lower the G-force to collect the yellow beads first in an intermediate bag. And we harvest the yellow beads. As you can see on the rotator picture, there's a bag full of yellow beads, leaving the blue beads behind because they're of different sizes. Yellow beads are 6 microns and blue beads are 10 microns. And then we concentrate the blue beads first. And finally, we bring down the yellow beads to reprocess and reconcentrate them back into the sample bag, or you could harvest them in different separate bags. Okay. So overall, the rotator can be used for many different applications. We've done basic cell wash media exchanges. We've done even processing of iPSC spheroids aggregates, about 100 micron aggregates in the rotator. We've done microcarrier MSC separations. We've done LV supernatant clarification. So we remove mammalian cells from LV supernatant. So we've done a lot of different things with our early adopters. In fact, the rotator has been validated by early adopters and beta testers for over a year now. And we do have about 5 end users in our region, in the Asia Pacific region, that are planning to use it for early phase clinical trials. Okay. And these are some data that has been provided by our R&D. So we've compared viability and recovery of T cells, so basic wash concentrate of T cells. It's always above 95%. We've looked at, achieving how much ability to concentrate down to low output volumes of 5 mils and achieve high cell density at the same time. And finally, we looked at recovery of PBMCs from washing leukopak. So we would have a fresh leukopak sample. We'll hook it up onto the rotator and we'll just do wash. And we were able to recover 88% on average, sometimes higher, sometimes lower, depending on donor variability. And typically, we only see 3% or less than 3% RBC contamination. Okay. In terms of the quality of cells that has been processed in the rotator, we've done an assessment processing T cells, and we looked at T cell composition of CD4 and CD8 T cells before and after rotator, compared with manual methods, and the composition is the same. So processing cells in rotator does not impact cell composition. And we also looked at broader cell types. So we process leukopaks, fresh leukopak, split this into 2 different experiments. We've done a comparison using a manual phycol. Manual phycol, we looked at different cell populations, and then we've also done with the same donor processing the fresh leukopak on the rotator system, which without any phycol, which took less than half an hour, and the cell compositions remains the same. So in other words, the rotator, we find that we can process fresh leukopak samples much faster than as opposed to using manual phycol separation. And overall, in the Rotier system, the cells are safe and secure. So this is a picture of a high-resolution camera, and these are cells in the chamber. Every run, you can view the live video. You could record what's going on in the process. And if anything happens during the process, you could pause the system, or you could stop the system. The pinch valves will go up, and the cells will still remain closed. And you can insert some recovery steps to recover your cells, just in should something happen. And we also, the software is open architecture. So you could make changes and adjust settings in real time during process development. And when it comes to translating your work to a GMP setting, you can lock down manufacturing, you can restrict user access, and you can ensure that the end users are not able to make any further changes in the run in a GMP setting. And finally, how does the CTS Rotier system enable users to comply with GMP requirements? First, the instrument itself is manufactured according to ISO 13485. And the Rotier system, we must clarify here, it's not considered a medical device. It's a cell processing equipment, and we intentionally didn't want to label it as a medical device because we want to give users the flexibility to use the same instrument for different parts of the process. The Rotier system is an automated closed system, and its intended use is either for research or for cell and gene therapy manufacturing. The single-use kit design will have its own certificate of analysis and COO with all the different types of tests and qualification that has been done according to USP guidelines. An example, you would want to test the plastics and have information on particulates, extractables, and leachables. So the kits will come with that documentation package. And finally, software. As I mentioned earlier, the system itself is not 21 CFR. However, it has a communication protocol called the OPCUA, which is an industry standard that can allow information and processes that goes on in the Rotier to be fed through OPCUA to your 21 CFR Part 11 compliant system, lab management system that you use in a GMP facility. And this is some of the early adopter feedback that we've got on the field. So Dr. Rebecca Lin is our collaborator from the Hudson Institute of Medical Research. She's probably the first adopter of Rotier, and she commented that Rotier is a flexible multi-purpose tool, and it's like the Swiss Army knife of cell therapy. And we have probably over 20 global users today. I think the most common feedback that we get on Rotier is that it's able to it's able to do so many different processes in the system, without having to, pay extra fees in protocol development. So we give our end users and customers the ability to make their own protocols, and there's a lot of flexibility. You could process any different cell types, and in some cases, this could be applied for even clarification of viral vectors. So together with the Rotier, what we are planning to provide is also compatible bags and media in bags. So next month we will be launching, basic CTF branded DPBS and IM5 media, which is a very well-established immune cell and T cell expansion media in the field today. So these are 2-liter formats, and it comes with tube weldable tubing that's compatible with the Rotier kits. But you would also be able to use any types of bags with the Rotier, as long as whatever bags that you're comfortable with. But also part of the our technologies that we offer on top of Rotier, we have T-cell isolation, Dynabeads, that has been already used in commercialized CAR T therapies along with our CTS Dynavac for isolation. And in the future, we are looking to address a market need in the area of scaling up electroporation. So today we have the Neon, which is commonly used in academic setting. You can only electroporate small volumes, about 10 microliters up to 100 microliters. But we do see a demand for electroporation in larger volumes. So we intend to provide a CTS electroporation system that could handle up to 25 mils of sample for electroporation, and this would be a closed system. And finally, in terms of expansion, we have different types of CTS branded media, and another common xeno-free, serum-free media that's been adopted in T-cell immunotherapy space is CTS optimizer media, together with the CTS immune cell serum replacer. So the immune cell serum replacer replaces the need to use human serum, and it's again, it comes along with the appropriate traceability documentation and regulatory documentation that's needed for clinical trial filings. And we've also validated protocols to upscale T-cell expansion in bioreactors on Rocking platform. So this is an overview of our vision for cell therapy manufacturing, and we plan to integrate different closed systems and modules and provide standardized protocols for end users, starting with autologous CAR T therapies. And Thermo Fisher, we remain very invested in the cell and gene therapy space. So we continue to develop innovative technologies, not only in-house, but explore partnerships with academic and industrial collaborators. And we're also investing in our CDMO capabilities and supporting GMP manufacturing of not only reagents, but some commercialization of cell therapies. So finally, we are here to help. So we have plenty of resources to guide you through the technologies that we have in cell and gene therapy, but also some information on and guidance on what you can look for if you're starting up your processes. And also we have another initiative called Ask Our Scientists. So we have a form online. If you want to reach out to our global or regional R&D scientists, you could fill it up and we'll set a time for you to have a specialized one-on-one consultation with our group of scientists. And finally, the Rotier, it's available for demos, and it will be launched in the next 1 to 2 weeks. We have demo units available in most countries in Asia Pacific. So I would highly encourage you to reach out to us for a demo. Okay, so so thank you very much for your time. I've now concluded my talk, and I'm just ready to take any questions.
Thank you, Jacqueline, for your talk. Give me a minute. Sorry. Okay, thank you so much. Thank you so much for your questions. We have quite a number of questions coming in, and Prem has been addressing them. So I'm just scrolling through and seeing if there are any other questions that are not answered. A question on the order lead time for instruments. What is the lead time on the instrument and lead time for the SUS items?
There is no impact of lead time for instruments. If we are talking about Rotier, we have them ready in stock. I think it would be available in anytime within 2 weeks.
Thank you, Jack. And a question on SUS. Not sure if that means single-use kit. Order lead time for SUS. I'm sorry, I may not know what this means.
I'm not sure. Perhaps the question could be clarified.
Yes. Wei, thank you for the clarification. I just saw your response.
Okay, sure.
Single-use.
Right. The single-use kit for the Rotia, again, it's available in inventory. Our typical shipment time is 7 working days. Now, I would say that two weeks is a comfortable time for shipment.
Okay, and also a question from Wei. Will Thermo Fisher support assembly of customized single-use equipment for?
If you're talking about Rotia kits specifically, at the moment, we are not supporting customization capabilities on Rotia kit, but we might consider that in the future. So it depends on the kit design will not change. The design is locked in. If you're talking about customizing the kit to pre-attach bags, that is something that we would consider in the future, but not at the moment as we have just launched the products. However, other single-use items such as our BPC containers, so our bioprocessing bags, we could customize them. So I think we might need a little bit more details on what type of single-use items you are looking at, because that really varies.
All right, thank you, Jacqueline, for the answer. I think today we will have to get in contact with you separately to address your questions. Thank you. And I don't see any other questions coming in, and I think we're right about on time, so I'm going to pass over to Sonia for the poll. And if you have any questions that you didn't manage to ask, you can also ask it in the last question of the poll. All right, thank you so much, and over to you, Sonia.
All right, thank you, Lydia. Just a quick check, can you hear me?
Yes.
All right, great. Hello, everyone. I'm Sonia again. So this will be our final poll for GGT Gibco Galaxy Tour 2. So everyone, just look towards the right side of your screen, and once you expand the tab, you'll be able to see the final poll questions. For this final poll, we'll be having 5 questions. All right, okay. First question, what cell types are you working on? So for this question, you can select more than one option. All right, so this is not a multiple choice, so you can select more than one. So we would like to capture your information as accurate as possible so that we can follow up with you closely. All right, for second question, which stage of development of cell therapy are you currently at? Are you starting out, or you have not started out, you are still looking around? So please let us know as well. Okay, and then for question 3, what are the key challenges faced during your cell therapy manufacturing? So for this question, you may also select more than one option. Is it labor intensive or having low cell recovery and viability? All right, so that would be for question 3. And for question 4, we are just keen to check out what are the products that you might be interested in. And the following options are all our current Thermo Fisher products related to cell and gene therapy as well. So for this question 4, you may also select more than one. So it's not multiple choice. So please fill in as many answers which are relevant to you. And of course, for the last question, if you would like a copy of presentation or you would like a demo for Rotia, right, that Jacqueline has presented earlier, please let us know so we can follow up with you very closely. And of course, for Southeast Asia researchers, we'll work with distributors and to arrange a meeting with you, either virtually or via a live demo. Okay, and if you might be interested in this product in 3 to 6 months' time, please let us know, and then we can follow up with you after that. All right, so I think if there's any other feedback, either regarding to the entire Gibco Galaxy Tour event, please let us know as well. Or if you think that there's other topics that you think that we can cover next time or for next year, let us know, and then we can put this into consideration when we prepare for the event. And I understand that many of you are also keen to have the certificate of participation. Yeah, so for those of you who attended our Gibco Galaxy Tour 1, which covers cell culture and transfection, and also attended our Gibco Galaxy Tour 2 for cell and gene therapy, you can, of course, get a certificate of participation. So just indicate in question 6, and then we'll follow up with you on this. All right, so these are the 6 questions that we have for this final poll. And if you complete all 4 polls, of course, you will stand a higher chance of winning the lucky draw, right, which is on the screen right now. You can see the Logitech headset and the UV box wireless charging. And for Southeast Asia customers, we'll take some time to actually send a gift over to you. And for today, we will not be doing a live draw. So instead, we'll be announcing the winners in 6 days' time. So you can check back to our website after that. All right, we have 2 minutes left for the poll. So for those of you who are still working on it, you have 2 minutes left. For those of you who have completed, I'd just like to share that a quick one to give you guys an overview. This might be more relevant for our Southeast Asia researchers. Okay, so for these are our distributors in Southeast Asia. So just a quick recap, for Malaysia, we have, of course, the Biodiagnostics, our key distributor over there, and of course, we also have the Fire as well. All right, and for Vietnam researchers, don't forget your go-to will be Vitech. And then for Indonesia, definitely it will be Nutrilab Pratama Key. All right, and of course, we also have researchers from Laos and Cambodia. So for Laos, go to Great Pharmet, and for Cambodia, we have a Unique Medical Distribution. All right, so these are our distributors from our Malaysia, Vietnam, Indonesia, Laos, and Cambodia. All right, in the next slide, for Southeast Asia researchers only, for Thailand, of course, I think you guys, your go-to distributor will be Thai. So if any questions, do reach out to Thai regarding any of the presentations today, and then we can follow up accordingly. And of course, FKN Laos, we have Delta Science for Myanmar, and of course for Brunei, we have COSI. All right, so these are distributors from Thailand, Laos, Myanmar, as well as Brunei. All right, so this will be relevant for the Southeast Asia researchers, and maybe I'll just do a final recap again. These are the distributors from Malaysia. BioD is the Fire, Vitech, of course, Vitech for Vietnam, Nutrilab, and our distributors from Laos and Cambodia as well. And of course, for Thailand, we have Thai. And for Myanmar, Delta Science, Brunei, COSI. All right. So we'll also be sharing these details with you guys post-event. So you guys can take a look and follow up with distributors accordingly. But if there's any other questions, please reach out to us. We are still here in the Q&A session. Jacqueline will still be around, and before we proceed to the Mandarin session later for the Taiwan session. So if you have any other burning questions for Jacqueline and our panelists, please let us know in the Q&A box to ask about Rotier or any other questions related to your field. All right, last but not least, just for everyone who's going to drop off soon, we'll be announcing the winners for the lucky draw on 14 October. So please go to thermofisher.com/GGT2020 to take a look, and then the winners will be announced. All right, okay, that's all for Gibco Galaxy Tour 2. And thank you for joining us at Gibco Galaxy Tour. We are very fortunate to have so many loyal fans of Gibco and supporting Thermo Fisher as well. We hope that you guys can continue to support us, and we hope that we'll do our best to help you to accelerate and advance your research in cell and gene therapy field. All right, so for those who are going for the Mandarin session, we'll see you at 11:30 a.m. Thank you. All right, thank you everyone for joining us at Gibco Galaxy Tour 2. And for those who are joining for the mentoring session, it will start in 15 minutes time. And for Southeast Asia researchers or Singapore customers, if you would like to stay on, please go ahead as well. Yeah. If not, please have a good day. All right.
Hi everyone, welcome back and welcome to our third and last session for today's Gibco Galaxy Tour. I hope you enjoyed your break. If you've attended the last session, which just ended about 15 minutes ago, please note that this is a repeat of the same session that was presented by Jacqueline earlier, and this will be presented in Mandarin by Dr. Jasmine Sung. I'll now like to invite our host for the second session, Dr. Claire Wu, our Regional Market Development Manager, to introduce our next speaker. Over to you, Claire.
线上各位大家好。那接下来的演讲呢，也是我们今天这个线上研讨会最后一个演讲。那希望就是大家如果有参加之前的演讲的话，希望你们已经有好好充分休息，然后准备好听接下来的这个演讲。那如果没有的话也没关系，那就敬请期待接下来用中文演讲的这一个新产品发表会。那接下来的这个演讲呢，是由我们台湾的产品经理 Jasmine 来为大家做演说。那这个内容的部分呢，就如同刚刚 Lydia 说的，就是内容的部分基本上是跟前一个用英文演讲的内容是一样的，那只是这个部分是由中文来做演讲。也因此呢，就是大家在演讲的中间如果有任何问题的话呢，都欢迎用中文在Q&A 的那个框框里面就是做询问。那我们会有专人就是在演讲的中间呢，用文字的部分做回答，或者是演讲之后 Jasmine 会用中文为大家做解释。那请 Jasmine 开始你今天的演讲，谢谢。
OK 可以听到我的声音吗？声音还 OK 吗
？Jasmine 可以听到你的声音，但是需要稍微大声一点。
大声一点。
对，有一点小。
谢谢你。
这样可以吗？
再稍微大声一点。这样可以了，你的笑声是刚刚好。谢谢你啊。
好，谢谢。好，那很谢谢刚刚 Claire 的介绍，还有 Lydia 的 opening。各位线上的听众大家好，我是Jasmine。那今天呢，很谢谢大家来参加这个线上的 webinar。接下来是整个 GT 系列最后一场的演讲。那我们的题目呢是应用于细胞治疗的制程开发与商业化量产的创新封闭式系统，也就是我们最新要推出的一台机器，它叫做 CDS Rotia Control Flow Centrifugation System。那接下来呢就会简单的跟大家做一个新品上市的一个介绍。首先在Cell and Gene Therapy 这个产业细胞与基因治疗的这个产业里面，其实是会遇到各式各样的一些挑战。那我们跟很多的研究者也好，或者是生技公司的研究者去做讨论以后，都了解到说，大部分大家会遇到的有以下的4项哦。那首先是 quality 跟 logistics， 就是说品质以及物流的部分。那如果各位有参加早一点的session 的话，可能可以听到我们的同事Mary Kay 有提到说关于环境的管控啊 QC 的部分要如何注意。那如果有参加到昨天的 session 呢，事实上我们也有好几位同事提到 QC 的部分，包括说去对你的细胞产品做一个确效或者是纯度的检测，以及原放标准的一个检测等等，这都是大家非常要去考量的部分，以及包括供应链是否能够完整或者是及时的一个deliver。那当然法规的部分也是非常重要的必须要有足够的文件啊，还有一些检测以及适当的一些 tracking 的记录来 support 这个部分，达到法规上面的要求。还有在一些风险管控上面也是要注意的。那也因为这个领域它的特性市场的变动其实是非常快速的，那一些创新的技术也一直的不断的推陈出新，所以怎么样在这个过程当中保持自己的就是这个创新度，或者是说保持在领先的地位，其实对大家来说也都是一个挑战。那我们今天要比较讨论的部分，其实是比较 focus 在第4个部分，也就是成本我们希望说在 这一个领域里头协助产业界在 scale up 或 scale out 的过程当中，能够降低细胞处理所需要的成本。那这个主要是透过采用封闭式然后模组化的一个系统。那最后呢，还可以利用数位化的方式，把这些系统都串联起来，然后达到真正的一个自动化。这张slide 呢的左手边其实是一个很典型的做autologous T cell 的细胞治疗这样的一个 workflow。从病人身上取得的这个血液样品呢，经过白血球的一些处理，那可能会需要经过isolation 把它分离，那去做 activation。那如果说要做 CAR 的话，你可能还要再 deliver 你的 CAR 进到细胞去做一些基因的编辑，增强它的毒杀效果或者是转移性的部分。那接下来会进到expansion， 然后养完了之后 harvest 下来，最后当然会需要做一些cell wash， 或者是说最后的分装到适当的一个保存的液体里面，那才送回到医院去回输到病人体内。那这样子的一个 workflow 其实跟我们在台湾的特管办法所规范的也是很类似的，把 cell 的 manufacturing 跟治疗的部分切分在2个区域里头。那在这样的过程当中，你可以了解，或者是说蛮多的人在使用的时候，其实是会从实验室或是学术上面使用的习惯这样来做。那其实中间就牵涉到蛮多就是手动操作或者是人力辅助的部分，那就很容易会遭遇到一些可能的问题。那右边列了好多项。在这里面来说，主要有几个点，一个是人力的支出成本可能会挺高的，那既然是手动操作，也就不免有可能会造成一些人为疏失，或者是说potentially 有一些 contamination， 有些污染的一个状况发生。那另外一层来讲呢，手动操作的话，可能中间会牵涉到，比方说离心机，比方说你在那个细胞的安全操作柜里面。但是这个过程当中，其实你很难去做一个适当的一个追踪，甚至于记录怎么样去使用，可能也没有办法有一个系统或者是软体的方法去这样子串联起来，这都会造成一些困扰。那另外呢，还有一个特点就是大家都不希望发生到一些所谓失败的一个状况，因为如果失败的话，意味着可能这位病人他就没有办法接受好好的就是细胞治疗的这个疗程。那对于一个正在进行clinical trial 的生技公司来说，这也是一个很大的影响，因为这意味着你达到你的 milestone 的这个时程可能就会延迟。那我们都理解到说这个部分其实是会造成大家的困扰。那一个 solution 就是希望说可以有封闭式，然后模组化的一个系统，可以保留操作上面的弹性，那也可以套用到自动化的功能来减少人力的支出，还有人为的疏失等等。那从这个成本的一个角度来看当我的整个生产制造的环境如果采用的是一个开放式的，然后手动操作的这样子的一个系统的话，其实就会需要在所谓 Class A 这样子的一个解禁室里面去控制它的落尘量或者是空气中的一些微生物这样的一个数量，那其实就是非常花钱的。不只是你要建制这样，你还要花费费用去做一个监控管制一个部分。但是如果我们采用的是一个密闭式的，然后自动化的这样子的一个系统的话，其实这个洁净室的等级就可以不需要拉到这么高，我们只要在Class C 这样子的一个洁净室就可以符合一个所谓的法规的一些要求。那这样子来说的话，以成本的角度来看，其实光是从 Class A 降到 Class C， 它就可以省下大概50%，大概一半的费用，在于这个仅仅只是在于环境的监控而已，所以还不包含这些建制的成本，是事后的监控。那更别提还有其他人力成本啊，或者建制成本以及清洁成本之类的，所以这其实是大家很值得考虑的一个部分。那其实这也就是我们Thermo Fisher 他想要提供给各位业界的使用者 我们希望建立一个就是优于业界的一个模组化的一个系统，那可以又可以上下游的去做一些串联。那这个系统里面，除了说我们有一些新的创新技术跟既有的平台之外，还包含了很优质的试剂跟已经优化过的一些 protocol， 以及包括到这个最终结果或者是在生产过程当中的一些分析的一些仪器。那如果说我们可以像这样子把它全部都摆在 bench 上面一条龙摆好，事实上我们还可以透过一个数位化的系统，把它连接到Delta V 这样子的一个platform， 然后去达到真正的串联，然后跟自动化的一个部分。那接下来呢，我们就要进入到今天的一个主角，我们即将要上市的新的一台机器，它就是 CTS Rotea Counterflow Centrifugation System。那它是一台很全新推出的一个机器，专门为了细胞治疗的 manufacturing 设计的。它是用来做细胞处理这样子。那它是一个桌上型，然后封闭式的一个机器，那也可以达到全面的自动化，可以运用在细胞的清洗、concentration 浓缩或者是 separation 分离。那它当然有几个它的特点跟它的优势。首先就是它在操作上面非常的弹性及灵活，指的是我们在开发的过程当中呢，可以按照你的需求去设定适合的 protocol。那这个 protocol 可以是针对比方说 T 细胞或者是 MSC， 那或者是你要运用在细胞的 separation 或者是 wash 或者 concentration 都是可以的。那这也不需要额外的所谓的软体或者是额外的费用就可以达成。那在细胞回收率跟活性的部分是大家非常在意的。这个部分来讲，我们很容易就可以看到说大概有95% 以上的回收率跟这个活性的部分。那原因是因为我们运用了所谓counterflow centrifugation 这样子的一个技术，后面会比较详细的去介绍这一块。那它可以给细胞造成比较小的一些压力，所以可以提高它的回收率跟这个活性的部分。那还有一个很特别的地方是，它最后输出的体积其实可以非常的小。今天不管呢你的起始的体积是10 liter 或者是1 liter， 我们最后都可以把它浓缩到最少是5 cc 这样子的一个体积。那这样子的体积其实是非常适合所谓自体免疫细胞的治疗这样子的一个需求。那在这个过程当中，其实我们是可以按照细胞的大小不同，把不同的 population 给分开，比方说把 lymphocyte 跟 monocyte 给分开来。那再来，我们也理解到大家的实际上的进展，可能是先从 R&D 就是制程的一个开发开始，然后会走到商业化制造这样子的一个需求。所以我们的设计呢，其实它就是一个很弹性的一个使用。它运用的是封闭式的一次性使用的一个耗材的 kit， 那可以确保它里面已经是无菌操作。虽然说就是机器本身我们并没有特别要让它是所谓的medical device 医疗器材，因为毕竟它不是用来做治疗用，直接做来治疗用的一个仪器。那但是我们有提供一个界面叫做OPCUA， 它其实是可以把我们的 Rotea 跟你现在实验室里面既有的已经符合21 CFR Part 11这样子的一个系统去做一个连接，来达到这个commercialize 的这样子的一个需求。那它可以操作就是处理的一个体积其实是没有上限的，我们一般会建议说可以用到10 liter 都没有问题。那这边可以给大家一个 idea 哈，如果说我有7到10 liter 的 MSC 的细胞，我们大概在 Rotea 上面呢是花1到1.5小时就可以完成wash 跟 concentrate 这样子的一个步骤，所以对各位在实验操作来讲会非常的方便。大家可以看到 Rotea 呢，它主要就是3个部分。第一个就是机器本体，那刚有提到说它的操作性简单，然后有多功能。那还有就是一个 single use kit， 也就是一次性使用的这个 kit， 它是唯一的一个耗材。 那这所有的管路呢都是一个密闭式的，所以可以确保整个环境里面的这个操作的细胞在操作过程当中是处于无菌的一个状态，密闭式的一个状态。那以及呢它的一个控制的一个软体，那这个软体可以去监控整个的流程，同时它也有一个摄影机，好，那你可以及时的去监控说细胞流进流出的一个状态是不是按照我们的一个预期。那如果说在真正的industrial 就是已经要商业化量产的时候，基本上我们可以使用welder 去确保整个 tubing 跟你的带子衔接的时候的一个密闭性。但如果说我们其实还在前期R&D 的阶段，还在开发的阶段，也可以使用lever lock 的方式把你的 tubing 跟你的带子去做一个衔接。那目前的话，我们整个系统是已经有 CE 跟UL 的认证，那在这一块上面也可以放心的一个采用。那因为Rotia 它其实是有非常多的功能，刚刚有提到说它可以用来做细胞的 separation wash 跟 concentration， 所以它在细胞治疗的 manufacturing 过程当中，其实是可以应用在好多的不同的步骤里头。那下面的话就是一个举例啦，如果说在 CAR-T 的 workflow 里面呢，你可以运用在好几不同的一个阶段，比方说从 leukopak 要去分离比方说你的 PBMC 或者你的 immune cell，甚至于是说 concentrate 把它换到比较小的体积，方便你去做一个separation 或者是 isolation， 然后把体积再继续浓缩，甚至于置换 buffer 去进行这个CAR 的这个 delivery， 不管你是要用电传染的方法，或者是要用virus 的方式去做 delivery。那处理完之后，你也可以再继续用 Rotia 去进行buffer 的置换，换到它适合生长的 medium。最后呢expansion 完了可以把它收集下来，做一个最后的 wash， 还有concentrate， 然后把它放在final 的一个适当的一个 solution 里面，就会输到病人的体内。我们其实有如果刚刚大家比较早登录进来的话，可能已经有看到这两个影片了，它是分别在介绍说 Rotia 它的一个它的一些特点，还有呢它的一些工作原理。那如果说各位有兴趣的话，可以再跟我们联络，我们可以把这样的连接留给大家，或者是我的同事可能已经贴在就是 Q&A 的 session 里的那个 box 对话框里头，大家都可以去点选去观看这样子。那大家或许不是那么熟悉所谓counterflow centrifugation 到底是什么样的工作原理，所以这边呢我就大概的简单说一下。首先必须要把样品load 到这个 chamber 里头。那我们的 kit 刚刚大家可能或许有点看到它的设计呢比较不一样，它有一个锥形的一个 chamber。那首先把细胞呢从这个小里面的管子流进去这个 chamber 里头。那我们会设定它的流速，那确保它的速度足够快，可以把所有的细胞呢流进这个chamber 里面，而不会塞在这个尖端的一个部分。那在此的同时呢，这个 chamber 会旋转起来，有点像离心机那样子，所以它会产生一个 G force 的一个力量。那你可以注意到 G force 的力量跟这个流速它其实是相反方向的作用力，所以呢我们可以去调整这两个之间的力量，让它达到一个平衡的一个状态。当它达到平衡的时候呢，所有的细胞就会在chamber 里头形成一个所谓叫做fluidized bed 这样子的一个状况。那这个是什么概念呢？这个就是跟传统离心最大不一样的地方。传统的离心其实是会把细胞形成一个 cell pellet， 然后会在管子的最底下。那其实是会有点挤压到我们的细胞，给它一些额外的一些剪力啊，或者是压力。可是在counterflow centrifugation 里面呢，细胞只会形成这种 fluidized bed， 它好像是就是所有的细胞会非常的靠近，但它还是 suspension 的一个状态，所以说不会给细胞带来额外的一些伤害。这也是为什么刚刚提到说，用 Rotia 处理完之后呢，细胞本身的 viability 并不会受到太大的一个影响。好，那在这个阶段呢，我们可以进行细胞的 wash 或者是 buffer 的 exchange。那接下来我们可以去调整 G force， 可以看到说这边箭头变小了，也就是我把 G force 呢降低，就是调整转速。 那 G force 变低之后呢，力量不平均，所以就会把比较小的细胞从这个锥形的顶部的部分流出去。那这样子的一个步骤，我们把它叫做 illustration， 或者说这是 separation， 目的就是把比较小的细胞跟比较大的细胞给分开来。那因为我们的这个设计呢，所以它可以很清楚的，就是可以很快速的把小的细胞或小的 particle 很快的流出去。所以呢，假设我们今天是从leucopad， 然后把细胞 load 进来，光是这个 loading 的过程当中，就可以把你的像血小板这一类比较小的这些 molecule 很快的就会洗出去。所以光是这样的步骤，你就可以得到更纯的一个 cell 的一个 population。那我们可以再更细的来看一下这个 illustration 跟所谓 cell separation 这个部分。如果说我们今天想要就是做 illustration 或 cell 的separation， 我们就把刚讲的就是说把 G force 调低。那这样子来讲的话，就可以把它做分开来。我们这边的例子啊，就是蓝色的部分是比较大的细胞，举例来讲，可能就是类似 monocyte， 那大概就是介在平均来说10 micron。那lymphocyte 它比较小一点点，那我们用黄色的这个例子来当做一个代表。所以当我的 G force 降低之后呢，黄色的部分比较小的细胞，它就会从底部流出去，那就可以达到我们想要做的 separation 的这样子的一个目的。如果我们今天想要收的是比较纯的monocyte， 就是大的细胞，我们可以在设定的时候，设定说我要收多一点点流出去的体积，那我的大细胞的部分就会比较纯。那相反来讲，如果说我希望是比较纯的一些小细胞，我们可以去做这样子一个调整。所以这个部分其实是非常弹性的。接下来的话呢是把刚才的这个就是图片的说明呢，试着用一些更直觉或者说实物上机器它在操作的一个方法说给大家看。那在 go through 这个protocol 之前，或者这个 demo 之前，我想要给大家看一个短短的影片，显示说就是我们如何把Rotia 这个机器给 set up 起来，然后会 go through 一个很简单的一个 protocol。稍等我一下。OK， 在这个影片是要介绍怎么样子去设定 Rotia 的机器，那并且简单的去go through 一个 protocol， 先把电源线连上，然后把开关打开，把机器跟电脑连线上，然后呢就可以把软体打开，那点选这个 select protocol， 不好意思有点累，点到 browse。那就可以找到相应的 protocol， 比方说那个 cell separation protocol 这样子。那接下来是把 kit load 放到机器上面，先把袋子呢按顺序挂上去，然后再来呢是把kit 的部分放上机器 ，OK， 然后再把管子呢沿着那个蠕动 pump 放好，然后把管子沿着这个沟槽把它放好。OK， 接下来呢是把中间的这个锥形的部分呢放在正中间，那可以利用银色的开关去控制它，把它卡好。接下来呢要把每一个管子压进去这个所谓 bubble sensor 的一个部分，确定它都一一的放进去了。然后管子跟袋子呢之间并没有造成一些纠结，然后这样子我们就可以把这玻璃门给盖上。盖上之后，机器会自动感应到，然后去做一些简单的一些自我检测的部分。好，确定没有问题之后呢， 我们就可以点选 Invitrogen， 它会把一些检测项目列出来。那前面的4项都是机器的自我检测，如果没问题，它都是打勾的状态。那最后的2项是我们使用者自己要去确认的。那如果没有问题之后呢，它就会开始，你就可以去机器上面按下绿色的按钮，它就会开始执行刚刚点选的 protocol。好，那执行完毕之后呢，接下来就是要把 kit 从上面取下来。首先呢，要记得把所有的管路都加进，避免液体会回流。那确认之后呢，我们就可以把玻璃门打开，然后呢，把中间的 chamber 拿出来，一样是利用刚才那个银色的开关。所有的管路拉出来之后，就可以整个把 kit 跟袋子拿出来。这大概就是整个使用操作的一个过程。好哦，那希望这样子大家可以稍微的了解一下怎么样去做设定，然后把机器的部分架起来这样子。那回到我们刚才的画面哦 ，PPT 的部分，你可以看到在这个照片上面啊，我们就是把3个袋子挂上去。以这个 bit 的 demo 来讲呢，最左边这个部分是一个wash 的solution， 然后中间是一个 intermediate 的 bag 是空的，那右手边的话就是一个 bit， 包括刚刚讲的蓝色的比较大的 bit 跟黄色比较小的一个 bit。那因为它是蓝色加黄色，所以看起来会是绿色的。那当我 launch 的这个 protocol， 你可以看到说袋子跟管路是要怎么样子的衔接，所以你完全可以按照这个指示去架设你的机器，就比较不会造成一些误差或者错误。当我开始 run 的时候，它第一个步骤是去做 priming， 也就是会先从这个buffer 的袋子里面把 buffer 冲进来，所有的管路之间就会充满了 buffer， 不要有空气存在。接下来呢，它就会有不同的阶段，所以它会显示说现在是从哪一个袋子流出来到哪一个管路。这样子的一个设计的好处是你随时可以去监控说到底有没有按照你设计的方法去进行。那这样子的 priming 其实大概在1分钟以内就完成。那所需要的 buffer 大概是40 liter 这样子，40 ml， 40 cc 就可以完成，所以很快就可以进入到下一个阶段。下一个阶段就是把 bit 给 load 进来。那这个时候呢，其实 camera 在机器里面啊，这个地方黑黑的地方其实是有一个 camera， 有一个摄影机，所以你可以及时的在这里看到sample 就是你的细胞或者 bit 流进来的样子。蓝色跟黄色都会流进来，同时你也可以在这边在管路上面看到它流进来的样子。那下一步呢，它会开始去调整它的 G-force 跟那个 flow rate， 你可以发现到说蓝色跟黄色渐渐开始做分层。好，那稳定了之后，我们就要做 illustration， 也就是把黄色的比较小的 bit， 或者是说我们模拟lymphocyte 的部分从底部流出来。这时候你在实际上可以看到机器这里黄色的 bit 就慢慢的流到中间的这个袋子里头了。接下来 chamber 里面呢就会只剩下蓝色的部分。那蓝色的呢就是模拟 monocyte 的部分。那确认之后呢，其实我们也可以一样调整它的转速跟流速，然后把这个大的 cell 给收下来。那你实物上就会看到说蓝色的 bit 流到这个袋子里面，所以我们就成功的把蓝色的bit 大概10 micron 左右模拟 monocyte， 跟黄色的 bit 大概6 micron 模拟lymphocyte 的 bit 给分开来。那接下来我们会把黄色的部分呢再进一步的去做一个纯化，所以再把它流回这个 chamber 里面。那同时也可以看到它在这里经过流速跟速度的调整之后，我们可以再把黄色的illustrate出来。那在实物上当然我们要分离细胞，一定是把它放在不同的 bag。那因为我们这是一个 bit 的一个 demo， 所以它又回到原本的 bag， 然后所以你又看到它变成绿色的。这样就完成了一个很简单的一个protocol 的部分。 看完了刚才的 video， 还有这样子 go through 的 protocol， 其实大家可以理解到这一台机器它有非常多的不同的运用的可能性。那这边其实列了一些我们很常用，或者说目前已经有一些客户有尝试过的一个做法，包括说 cell wash 啊、concentration、nutrition 等等之类。在我们的early access 的一个客户里头，他们有做过很多不同的一些尝试。所以在这样子的一个过程当中，其实可以运用在cell therapy 的 manufacturing 或者是 cell processing 的各个不同的一个步骤里头。那除了说它操作很方便呢，也是可以分离细胞或达到我们想要做的目的之外，大家当然会很关心说回收率还有 viability， 就是细胞活性的部分。这边就有做一些实际的一些测试，你可以看到说假设我们用Gibco然后去 recover T 细胞的部分，在 wash 跟 concentrate 完之后，它的 viability 跟 recovery 其实都可以超过 90% 以上。那再来中间的这个图像，在讲的是我们从一开始 input 的时候是1 liter 的 cell， 我们把它浓缩到最后，其实只剩下5 cc，OK 的200倍的一个浓缩。但即便这样子的处理过之后，它其实的回收还是可以达到非常高，回收率是 97%， 那 cell 的 viability 也是跟原本处理前其实是维持相当的一个状态。那主要是因为刚刚讲的control flow centrifugation 给细胞带来的压力其实是比较小的。那如果说我们拿Gibco的 cell， 然后要 recover 里面的 PBMC， 事实上我们就可以得到就是89% 的一个 PBMC 的一个population， 那里头剩下的 RBC 就会非常的少。那当然这个部分的话是要搭配RBC lysis buffer， 就可以处理有这样的一个处理的步骤。另外的话是这样子的处理虽然得到很高的viability 跟 recovery， 那是否会改变里面的T 细胞的一个组成呢？我们一样来做这样的一个分析。主要是在Rotia 处理之前跟处理之后，你可以发现到说在 CD3 positive 的 T cell 里面呢，CD4 positive 跟 CD8 positive 的 population 这个比例其实还是维持在相当的。所以意思就是说，经过 Rotia 的这个处理，它并不会好像只特别分到CD4 positive 或者 CD8 positive 这样的 T 细胞，还是维持原来的一个比例。我想这是大家会比较希望看到的一个部分。另外呢是做一个 PBMC 在做 separation 的一个比较，大家最常用的方法应该就是用 Vitech， 所以呢我们就拿了一个新鲜的一个Gibco， 从一个病人身上得到的 Gibco， 那我们把它分成一半呢用 Rotia 来做处理，一半就是用传统 Vitech 离心的方法去处理。那用 Vitech 的话大概是花费2个小时，然后呢得到了这样子一个 separation。Rotia 大概是不用30分钟就完成了，可是我们可以来看到说，它不管在NK 跟 T 的 population 里面，T 细胞跟T 细胞跟B 细胞或 NK 其他 monocyte 之间的 population 与用 Vitech 处理其实是非常相近，不一样。那B cell 跟 T cell 的这个 population 里面呢，其实leukocyte 跟 RBC 的比例跟 Vitech 也是非常的相近。那在于 monocyte 跟 T cell 的这个 population 来讲，因为我们可以把platelet 这个血小板的部分清洗得很干净，所以细胞本身的占比就会比用 Vitech 来讲会来得高一些，所以可以在比较短的时间内得到相同结果的一个细胞的population， 这是这张图来想要说的一个部分。另外呢，大家刚刚可以看到，其实整个是一个密闭的一个系统哦，所以你的细胞在 Rotia 上面处理的时候，其实是非常安全的。所谓的安全的意思是说，它不会就不见了，或者是掉到哪里去。一个是我们刚刚的 camera 可以及时去监控细胞的一个 processing 的状态。那如果说万一有发生任何的一些临时的状况，或者是不预期的状况，其实细胞一个是会在chamber 里面，在管路里面，或者是在袋子里面，你随时都可以暂停或者是调整转速跟流速，然后把细胞给回收回来，所以永远是不会说就不见掉，因为它是一个 closed system。 那我们要怎么样去 support GMP 的 manufacturing 呢？其实呢，在软体上面有个功能，如果是在 R&D development 的一个阶段呢，我们可以在 run 的同时，随时去调整这些参数，立即的看到它的效果。但是当我们走到要 commercialize 的时候，为了要符合 GMP 的一个精神哦，我们其实是可以把这个 protocol 给 lock 起来，给锁定，所以操作的人是不能去跟动这个部分，会去比较符合 GMP 这样子的一个精神。那除此之外呢，我们的仪器本身的制造都是符合 ISO 13485这样子的一个规范。虽然它不是medical device 医疗器材的一个角度，但是呢在于我们的 single use 的 kit，它其实是一个封闭式的这样子的一个设计。那生产制造当然是符合 ISO 这样的一个规范，以及有做很多的一些安全性的一些检测。那同时在软体的部分，我们也有提供一个使用界面 OPCUA， 它可以把我们的 ROTIA 跟你们实验室里面既有的符合21part CFR Part 11这样子的一个仪器系统给做连接，所以你可以更好的去追踪以及记录这使用的过程。我们的 Early Adapter actually 在全球来讲已经有大概20多个客户。从1年多前吧就开始在使用我们的 ROTIA。那Rebecca Lim，Professor Rebecca Lim， 她是其中的一位使用者。她其实觉得说ROTIA 它的功能非常的弹性，也非常的多元，就好像是个瑞士小刀一样，可以提供我们各式各样的一个运用。那我们的客户也会觉得好像因为它太方便了，很好操作，所以都很希望说把各个就一直去想说，我这个过程当中有什么步骤还可以在 ROTIA 上面进行。那给各位参考一下，在我们 APJ region 其实已经有5个early adopter user， 他们开始在使用，也都有进入到一些preclinical 或是 clinical 这样子的一个运用。后面的话呢，使用的 medium 当然我们没有任何的限制，只是说CDS M5 呢，它是一个很适合用来培养immune cell 的一个 medium， 那它有一个 bag 的包装，所以这个袋装的一个 medium 它就可以很容易的衔接上我们 ROTIA 的一个系统。那我们即将要上市的是一个 CTS 版本的 PBS， 也是要有袋装的。那当然如果说您有自己偏好的一个袋装的方式，其实也都可以跟 ROTIA 这样连接上使用。那刚刚一直有提到 CTS， 其实这个是一个叫做 Cell Therapy System 这样的一个缩写。我们所有的产品呢，如果说有符合以下这几个要点，我们就会把它然后是适合细胞治疗这样子的一个过程，我们会把它挂上 CTS 这样子的一个品牌，表示说呢它是符合 CGMP 的 manufacturing， 甚至于说 USP 1043的一个要求。那在产品上面本身也会做一些额外的一些检测，还有完整的一些 documentation， 一些文件、一些记录来 support 我们的客户。而且它也被运用在很多的临床上面的 clinical trial， 甚至于是 FDA 已经核准，欧洲已经核准的 CAR-T 这样的 therapy， 也都有使用我们 CTS 的产品。全球来讲，应该已经有超过200个以上的 clinical trial 都是使用我们 CTS 的产品。那包括说像在做 T cell 的isolation activation expansion， 很多人会使用 CTS 的Therapies 来做这个部分。目前应该是有蛮多 clinical trial 跟甚至于已经被核准的一些临床治疗是使用这个 kit， 然后搭配它的词作，它可以 handle 到最多是10 liter 这样子的一个sample volume。那如果说要做genome editing 呢，除了像昨天我们有Craig 讲到的VMAX 这样子的系统去做之外，用 virus 的系统之外，也有人可以用电转的方式。目前我们有的是 Neon 的 electroporation， 但是它现在能够处理的细胞呢，大概是10到100 microliter， 可能不是那么的够用，所以我们即将要推出的是或者现在正在准备的是 CTS 版本的一个 True Fluorescence System， 它可以处理到最高25cc 这样子的一个 sample。那 expansion 跟 scale up 呢，除了说一般的操作之外，我们也可以搭配使用到 bioreactor， 所以量产也不是一个问题。我们 Thermo Fisher 其实是非常希望在这个 cell therapy 的这个领域里面呢，跟产业界或者是学术界有一个 partnership。 那这个部分我们也很愿意去投资，跟大家的这个 partnership， 透过这样子的一个关系去创造出新的技术，甚至于说为了客户的需求，我们可以增加我们 CGMP manufacturing 的空间跟这个 capacity 来 support 我们的客户。那最后要跟大家分享的是，我们在网页上面其实有很多的相关的资源，大家可以去看一下。那要特别提出的是 as your scientist 这个部分，因为现在 COVID 的状况，那可能不是那么方便的邀请到我们国外的相关的专家们来到台湾，然后到各位的实验室去做面对面的拜访。所以如果说你有需求的话呢，其实是可以在这个页面提出申请，包括你有兴趣的主题，然后想要的语言 ，OK， 那我们就可以OK 可以去安排这样子的电话会议也好，或者是网络会议，那针对您的需求去做一个相关的讨论。那最后呢，跟各位再提醒一下 ，Rotia 其实即将在这个月的16号全球同步上市，应该是15号，但在台湾是16号。那所以如果说各位有兴趣想要多了解，甚至于说想要安排 demo 的话，都可以跟我们做联络，那或者是跟当区的负责的业务，或者是说写信email 给我都可以，那我们都很乐意跟你们做一些讨论。OK， 那我今天的 presentation 应该是到这边就结束了，谢谢大家的聆听。那如果有任何的问题的话，都欢迎在 Q&A 的那个 chat box 里面提出。
这个演讲的这个 section 结束还有一点时间，那如果大家有任何问题的话，都欢迎在右下角的那个 Q&A 的框点选一下，然后就会有个框框，你可以输入问题。那我们也已经放了一些就是跟 Rotia 有关的连结，那您可以就是就是点选那些连结，然后多了解一下 Rotia 这个产品。线上那个 James 有问一个问题，就是Rotia 可以用就是全血的 sample 当成是 starting material 吗
？OK， 主要是这样，全血里面呢含有太多的红血球。那个 chamber， 我们那个 chamber actually 它的体积大概是10 cc， 所以它能够就是容纳。虽然你觉得10 cc 好像不是很大，可是基本上它可以容纳非常多的细胞，从最小50个 million 1到5个 billion 都可以。可是红血球真的太多了，所以很容易就把那个空间给塞满塞爆。所以我们会比较建议说，使用 Leucopad 这样子的一个 sample 当做一开始的 input， 或者是说 PBMC。那事实上在真正 commercialize或者说临床上使用，你拿到的 sample 也不会是全血啦，也会是觉得对病人来说可能太辛苦了。拿到的可能是 Leucopad 这样子的一个 sample。
好，那如果就是线上的听众都没有其他的问题的话呢，那 我们就把时间交给Sonia 请她就是执行最后。
不好意思，好像有个问题跳出来。
好
，OK， 就是James 这边又有提到说，是不是有可能去分就是 culture media 里面的 exosome？因为 exosome 的 size 真的非常非常的小，太小了，所以它可能很快的就会被就流出去了，所以在这个部分可能还是没有办法做到这个部分。大家已经开始在想要怎样运用我们的 Rotia 了吼。
好，那谢谢 Jasmine 的分享。那么我们可以去看一下台湾经销商的部分吗？是呀，就是这样 Jasmine 可以说一下。
Sure。OK， 所以在线上的各位台湾听众啊，就是如果你们有兴趣任何的一个产品，其实都可以直接跟我们任何直销团队做联系，或者说在你们的区域里面可能有我们的经销商。那在新竹的部分呢，或台中的部分，就是新竹或者是像桃园就台北以南的区域呢，其实可以找我们在我们的经销商德仪啊。那如果说在中部地区的话，可以找我们的经销商是量子。那如果说是在南部台南、高雄、屏东其实可以找我们南部的经销商进阶，那都很欢迎你们跟他们做联系或者说直接跟我们做联系。那我们画面上现在秀的是他们的那个联系的 email address， 或者是说可以跟你们平常熟悉的业务联络都是 OK 的。
好，谢谢 Jasmine。我们还有什么问题吗？好的，如果大家还有什么问题，可以在那个 Q&A 的对话框发问 ，Jasmine 还有 Claire 都还会在线上。那么我们现在进行最后一个部分，就是 polling 的部分。那今天的 polling 一共大概有6个问题，然后大家可以往你们的右边看一下，那个问答题应该快要出现了。然后今天大家有5分钟的时间来完成这个问答题。大家你会有机会领取紫外线消毒盒还有裸体的耳机麦克风。